Welcome to Sleepy Time Stories. Hey dear friends, deep inside the story of human evolution lies a fascinating mystery written into our own blood. Among the billions of people on Earth, only a small percentage have Rh-negative blood, a rare genetic trait that has fascinated scientists, historians, and curious minds for decades. But why is this blood type so uncommon? Where did it come from? And what can it tell us about the ancient journey of humanity? For generations, researchers have studied the origins of blood groups, tracing the connections between genetics, migration, and human adaptation. Every discovery reveals another piece of the complex story hidden within our DNA. From ancient populations to modern science, the mystery of Rh-negative blood takes us on a journey through evolution, ancestry, and the incredible diversity of the human species. Before starting the story, like and subscribe to my channel for more stories. Now, start the story. Tonight, we'll explore what scientists know about this rare blood type, the theory surrounding its origins, and the ancient clues that continue to shape our understanding of human history. So dim the lights, relax, and join us as we uncover the fascinating mystery of Rh-negative blood and the secrets hidden within our own biology. There is a figure that appears time and again throughout certain fields of study, and the moment you notice it, it starts to linger in your thoughts, refusing to fade away. That figure is 15%. It is the approximate worldwide proportion of individuals who are Rh-negative. Depending on your location, that translates to roughly one person in every 6 or 7. Journey across most of Asia, and that number falls to practically nothing. Travel through much of sub-Saharan Africa, and it barely appears at all. In select areas of the Middle East, it hovers around 4 or 5%, occasionally lower. Yet when you shift your focus to the United States, an unusual pattern emerges. The number rises. It rises in ways that mainstream population genetics finds difficult to explain in a straightforward manner. It becomes concentrated within specific communities, particular lineages, and distinct regions that carry very ancient and very particular histories. As you begin to pull at that thread, as you start questioning why the American landscape appears the way it does when mapped against Rh-negative distribution, the answers that surface are neither simple nor comforting. Many of them have remained in plain sight for centuries, simply waiting for someone to pose the right question. If you are listening to this in order to drift off to sleep, that is precisely the purpose behind these creations. There is a unique quality to a mystery narrated slowly in the darkness, one that allows the mind to gently release its grip. You do not need to chase anything tonight. You do not need to solve anything. You can simply allow the narrative to pass through you, much like water travels over stone, gradually and with immense patience. Perhaps consider subscribing if you would like more of these, and feel free to share where you are at this moment and what time it is. I truly enjoy reading those comments. Someone in the Pacific Northwest at 2 in the morning. Someone in rural Georgia just past midnight. Someone in a city apartment who cannot quite articulate why they feel they are carrying something ancient within their veins. Before we proceed any further, we need to establish a common understanding. This is not a tale about superiority. It is not a story about a chosen few or exceptional lineages in the manner that certain corners of the internet like to present. Rather, it is a narrative about movement, about suffering, about population collapses and survivals that left their imprints on human DNA in the same way glaciers leave grooves in stone. It is a narrative about the peculiar, unusual, and largely overlooked history of a nation that drew people from virtually every corner of the planet, and about how that process of absorption created a genetic terrain that does not align with what conventional textbooks predict. Running through all of this, like a crimson thread embedded in pale rock, is this one specific protein, or more accurately, the absence of it. The Rh factor, in itself, is not particularly complex. It is a protein, It resides on the exterior of your red blood cells. If it is present, you are Rh positive. If it is absent, you are Rh negative. That is the entirety of the biochemical explanation. However, the consequences of that absence are far-reaching. They extend much further into the past than most people realize. They go back beyond the founding of the United States, beyond the colonial period, beyond the arrival of any European vessel on American shores. They reach into geological epochs, into the remote history of human migration, into ice ages and land bridges and a North American continent that was inhabited by human beings for a much longer span than historical accounts are generally willing to acknowledge. Here is the version of events that history books typically offer regarding America and Rh negative blood. European colonists began arriving in the late 1400s. The majority of Europeans carry Rh negative blood at rates between 15 and 20%, with higher frequencies in some Western groups. Those colonists established a nation. Additional immigrants followed, many of them also hailing from Europe. The United States, therefore, exhibits elevated Rh negative frequencies compared to much of the rest of the world because it is a country built largely from European genetic foundations. End of story. Move along. Yet that explanation is riddled with inconsistencies, significant ones. And addressing those inconsistencies requires venturing into territory that official history tends to avoid. The first inconsistency is geographic. Rh negative blood in America is not spread uniformly, as a simple narrative of European immigration would suggest. It is clustered. It is concentrated in ways that do not correspond to European immigration patterns in the slightest. Some of the highest concentrations are discovered in populations and regions with very specific backgrounds. Backgrounds that involve ancient encounters, genetic intermingling, and in certain instances, the deliberate separation of bloodlines over many generations. When you plot American Rh negative frequencies against the known patterns of migration and settlement over the last 5 centuries, the resulting map does not add up. Not without additional explanations that mainstream history is hesitant to provide. The second inconsistency is temporal. The prevailing assumption is that Rh negative blood arrived alongside the Europeans. However, DNA analysis of ancient American remains has uncovered genetic markers that significantly complicate that assumption. There are samples from pre-Columbian burial sites that carry unusual genetic signatures. There are populations in North America, some of them indigenous, others descended from peoples who arrived before the colonial period through routes that were not the transatlantic slave ships or the Spanish galleons, that carry Rh negative frequencies which cannot be explained by post-1492 European contact. And the third inconsistency is the one that most people prefer not to examine directly. It concerns what took place in the centuries following European contact. The deliberate relocation of human populations, the forced transportation of millions of individuals from Africa, from the Caribbean, from Central and South America, the devastation of indigenous communities, and the scattering of their survivors into new genetic pools. The formation of isolated communities in the American South, in Appalachia, in the desert Southwest, communities where genetic drift could operate on small populations over many generations, concentrating rare traits in ways that would never occur in large open gene pools. All of this contributes to why America's Rh negative map appears the way it does. And all of it is considerably darker than a straightforward immigration narrative would imply. So, let us begin at the very start. Not the start of America, but the start of the blood. The protein we refer to as the rhesus factor was named after the rhesus monkey, because the experiments that identified it in the 1940s used rhesus macaque blood to generate the antibodies that first detected it in humans. The name persisted, even though the actual protein in humans is not identical to the one found in rhesus monkeys. What is significant is this. Almost every primate on Earth possesses some version of this protein complex. It is ancient. It has been conserved across species. It fulfills important functions in the biology of red blood cells, and virtually every primate lineage carries it. Except in a small percentage of humans, it is simply absent. The gene responsible for the primary Rh antigen, the Rhd gene, is either deleted or inactivated in individuals who are Rh negative. This is not the acquisition of something novel. It is an absence, a deletion. And that absence behaves in ways that leave evolutionary biologists genuinely perplexed, because it persists at significant frequencies despite creating a very real reproductive disadvantage in certain situations. That reproductive disadvantage is something we will revisit in detail, as it is central to the darker reasons alluded to in our title. For now, simply keep this in mind. A trait that can cause complications during pregnancy, that can turn a mother's immune system against her own unborn child, has not only survived in human populations, it has flourished in some of them, including the American population. And the question of why this is so takes us into some of the most unsettling chapters of American history. But first, let us consider where Rh negative blood originated in the first place. The most ancient Rh negative lineages that we can trace genetically appear to have emerged in populations that inhabited Western Europe and possibly North Africa before the great migrations of the Neolithic period. Before agriculture spread from the Fertile Crescent and began reshaping the genetic landscape of Eurasia. Before Indo-European speakers arrived on horseback and altered the gene pools of much of the European continent. Before any of that, there were older peoples who left their genetic imprints in the isolated corners of the world where later migrations did not reach: the mountains, the Atlantic coastlines, the remote islands. The Basque people of northern Spain and southern France are the most renowned example. They carry Rh negative blood at rates approaching 35% in some communities. They speak a language that does not belong to any known language family. Their genetic profile is distinct from that of their neighbors in ways that suggest they represent a remnant population from a much earlier occupation of Europe. A population that was present before the agricultural revolution, before the Bronze Age migrations, before most of what we call European history. They are a living repository of something ancient, and Rh negative blood is one of the clearest indicators of that ancient lineage. Similar patterns emerge in other isolated populations. The Berbers of North Africa, highland populations in the Caucasus, the Sami of Scandinavia, the Aboriginal inhabitants of the Atlantic Islands. Wherever you encounter a community that remained isolated from the major migration waves of the last 10,000 years, you tend to find elevated Rh negative frequencies. And the theory that best explains this distribution is that Rh negative blood was once more prevalent across a specific geographic zone, and that later migrations diluted it almost everywhere except in these isolated pockets. What was that zone? The evidence points to the North Atlantic fringe, the western edge of Europe and North Africa. Populations living along that coastline from Portugal up through France, Britain, Ireland, and into Scandinavia show consistently higher Rh negative rates than populations living further east or south. And this is where the American story begins to connect to something much older, because that Atlantic fringe is exactly where the colonization of the Americas primarily drew its people from. The English settlers who founded the early American colonies came disproportionately from regions with elevated Rh negative frequencies. West Country England, the Celtic regions of Britain and Ireland, parts of northern France. These were not average European populations genetically. They were populations drawn from the Atlantic fringe, and they carried the genetic legacy of that ancient lineage with them across the ocean. But that represents only the first layer of the explanation, and it is the most benign one. The layers beneath it are considerably darker. The second layer begins in West Africa in the 1600s. The transatlantic slave trade brought approximately 12.5 million individuals from sub-Saharan Africa to the Americas over roughly 3.5 centuries. Most of those individuals were taken from coastal and near-coastal West African communities. And West African populations, like most African populations, carry very low rates of Rh-negative blood. In some communities, the rate is effectively zero. In others, it hovers around 1 or 2%. The trait is rare because the genetic history of sub-Saharan Africa does not include the isolated Western European lineages where Rh-negative blood appears to have originated and concentrated. So when those 12.5 million individuals were brought to the Americas, the expectation from a simple genetic mixing model would be that their descendants would carry very low Rh-negative frequencies, reflecting the low rates in their ancestral populations. And for a while, that is roughly what the data indicates. But here is where the history becomes dark enough that most textbooks tend to look away from it. The plantation system in the American South was not a static arrangement. It was a system that deliberately disrupted, mixed, and controlled human reproduction on an enormous scale over multiple generations. Enslaved individuals were bought and sold, separated from their families, moved between plantations, forced to reproduce with partners chosen not by themselves, but by the economic interests of the individuals who owned them. Children were separated from parents. Siblings were scattered. The genetic community that a West African village would have maintained over generations was deliberately shattered and reshuffled in the American South. In that context, the mixing of genetic material between European-descended and African-descended populations was not voluntary and was not symmetrical. The historical record is unambiguous about this. The rape of enslaved women by white enslavers and overseers was systematic and widespread. It produced children who carried genetic material from both lineages. Those children, classified as enslaved under American law regardless of their genetic heritage, were absorbed into the African American population, and the European genetic material they carried included whatever proportion of Rh-negative blood their white ancestors happened to have. This matters for the Rh-negative map of America in a way that is seldom discussed openly. The African American population today carries Rh-negative frequencies that are significantly higher than the West African populations from which their ancestors primarily came. Estimates vary by study and by community, but figures in the range of 7 to 10% are commonly reported, compared to the 1 to 2% in many West African populations. That difference reflects centuries of forced genetic mixing. The Rh-negative blood that European enslavers carried was transferred into the African American gene pool through violence, through coercion, through the systematic denial of bodily autonomy to enslaved women over more than 200 years. When people examine the American Rh-negative map and observe that it is higher than most other countries, they are in part looking at the genetic legacy of slavery. They are looking at what happens when European genetic material, including Rh-negative lineages, is forcibly introduced into populations that did not originally carry it over generations and at massive scale. That is one of the darker reasons, and it is not a comfortable one to dwell upon. But it is not the only one, and in some respects, it is not even the most enigmatic one. The third layer of the American Rh-negative story involves a population that is almost never included in these conversations: the indigenous peoples of North America. The standard assumption in genetic research is that pre-Columbian Native American populations had essentially zero Rh-negative blood. This assumption is based on the prevailing model of how the Americas were populated, which holds that the ancestors of all indigenous Americans came across the Bering Land Bridge from northeastern Asia during the last ice age, approximately 15,000 to 20,000 years ago. Asian populations carry very low Rh-negative frequencies, generally below 1%. So their descendants in the Americas should carry similarly low rates, and in many indigenous communities that is exactly what is found. Rh-negative blood is rare to absent, but not in all of them. There are indigenous and mixed heritage communities in certain parts of North America, particularly in the eastern United States, in parts of Appalachia, and in the southwest, where Rh negative frequencies are anomalously high given what we would predict from the standard model. Researchers who have noticed this anomaly have generally attributed it to post-Columbian admixture with European populations. But some geneticists and archaeologists have raised a more uncomfortable possibility, one that the mainstream academic establishment has been slow to engage with seriously. What if some groups arrived in North America through routes, and at times that the standard Bering Land Bridge model does not account for? And what if those groups carried Rh negative blood as part of their genetic heritage? The evidence for a pre-Columbian transoceanic contact with the Americas is more substantial than popular culture typically acknowledges. It is not mainstream science, but it is not fringe conspiracy theory either. There is genetic evidence of contact between South American populations and Polynesian peoples at least 1,000 years before Columbus. There is archaeological evidence of pre-Columbian Asian artifacts in North American sites. There is skeletal evidence from certain North American burial sites that does not fit the expected profile of Bering Land Bridge migrants. Remains with cranial morphology that more closely resembles certain European or North African populations than the Northeastern Asian ancestors that standard models predict. None of this evidence is conclusive. None of it proves direct transatlantic contact in the pre-Columbian era. But it raises questions that the standard model cannot cleanly answer. And those questions matter for the Rh negative story. Because if some ancestral populations did reach North America through Atlantic or other routes that passed through or originated in the regions where Rh negative blood is most concentrated, then some of the anomalous Rh negative frequencies in certain American populations might not be the result of European colonial mixing at all. They might be something older, something that was already here. That is a deeply unsettling possibility, both for conventional archaeology and for the political narratives that surround questions of indigenous American identity. It does not fit neatly into any of the existing stories that people like to tell about who was here first and where they came from. And that discomfort may be part of why it has not received the serious investigation it deserves. Let us spend some time with that possibility, because it leads us into a part of American history that most people simply do not know exists. The story of pre-Columbian North America is far stranger and far older than the standard narrative suggests. When European explorers first made contact with the indigenous peoples of North America, they found not a continent of scattered primitive tribes, but something considerably more complex. They found evidence of civilizations, some of them already in decline, some of them still flourishing, that had been building, trading, and developing complex social structures for thousands of years. The Mississippian culture, which dominated a vast swath of the American interior for several centuries before European contact, built cities. Cahokia, at its peak around the year 1100, had a population comparable to London at the same period. Monk's Mound at Cahokia is larger at its base than the Great Pyramid of Giza. It was built entirely by human hands, without metal tools, without the wheel, without draft animals. It was the center of a city of perhaps 20,000 people, a city with plazas and wooden palisades, and a hierarchy of social classes, and a trade network that stretched from the Gulf Coast to the Great Lakes. By the time European explorers reached the interior of North America in any substantial numbers, Cahokia was already abandoned. The great city had been empty for perhaps 200 years. Nobody knows exactly why it was abandoned. The theories range from environmental degradation to social collapse to climate change to warfare. What is known is that when the Europeans arrived, they found the mounds still standing and the local people still referring to them with a kind of reverence that suggested memory of a great civilization, even if the details of that civilization had been lost or deliberately concealed. And Cahokia was not alone. The Hopewell tradition, which preceded the Mississippian culture and extended roughly from 100 BCE to 500 CE, created earthworks across an area stretching from the Gulf of Mexico to the Canadian border and from the Rocky Mountains to the Appalachians. The Newark earthworks in Ohio, when complete, covered 4 square miles. 4 square miles of precisely engineered earthen geometry, circles and octagons aligned with the rising and setting of the moon across its entire 18-year cycle. This is not the work of small bands of wandering hunter-gatherers. This is the work of a civilization that understood mathematics, astronomy, engineering, and long-distance coordination. And then there is the copper. The old copper complex of the Great Lakes region produced copper tools and ornaments between 8,000 and 5,000 years ago. Not smelted copper, cold-hammered copper worked into intricate tools and objects by individuals who had figured out metallurgy thousands of years before the Bronze Age reached most of the world. The copper moved through trade networks that stretched across the continent. Copper objects from Lake Superior have been found in burial mounds in Louisiana, in Florida, in Kansas. Someone was managing a continental trade system, moving heavy goods over enormous distances, maintaining contacts across a geography the size of modern Europe at a time when the standard history of North America says there was nothing here but scattered bands of Stone Age peoples. All of this matters for the Rh- question for a very specific reason. Complex civilizations with large populations, long-distance trade, and sustained contact between different communities create conditions where genetic material moves, where traits that might otherwise remain isolated in one population get distributed through a network, where something like an elevated Rh- frequency in one founding population could over centuries spread through that population's trading partners and cultural descendants. If there were populations in pre-Columbian North America that carried Rh- blood, whether because they arrived through transatlantic routes or through some other mechanism we have not yet understood, and if those populations participated in the great continental trade networks of the Hopewell and Mississippian periods, then the trace of their genetic contribution could show up in unexpected places, in the descendants of peoples who never had direct contact with Europeans, in communities that mainstream genetics would not expect to carry this trait. And when Europeans did arrive, bringing more Rh- blood with them, that trace would be amplified. It would be mixed with and reinforced by the new European contribution, and the results several generations later would be the anomalous distribution pattern that researchers notice but rarely examine closely. This is a hypothesis. It is not proven, but it is consistent with the evidence, and it is more interesting than the simple story of European immigration that textbooks prefer. Now let us discuss something that nobody in the mainstream conversation about Rh- blood ever addresses directly: the role of isolated communities in American genetic history. America is a country that has for most of its history been characterized by mobility. The frontier mythology is built on movement, on people picking up and going somewhere new, on the restless relocation that defined the westward expansion. But within that story of constant movement, there are pockets, communities that did not move, communities that stayed in the same geographic area for generations, sometimes centuries, separated from the wider American genetic melting pot by mountains, by swamps, by social barriers, by poverty, or by choice. Some of these communities are famous. The Melungeons of Appalachia are one of the most studied examples. The Melungeons are a mixed-race community whose origins have been debated for centuries. They were present in the mountains of Tennessee, Virginia, Kentucky, and North Carolina before European settlement reached those areas in significant numbers. Early European settlers found them already there, already occupying the mountain hollows, already established, already speaking English or a form of it, already identifying as Christian. They had dark skin in some cases, lighter skin in others. Their genetic heritage, when analyzed with modern tools, turned out to be extraordinarily complex. Sub-Saharan African ancestry, European ancestry, Native American ancestry, and in some analyses, traces of ancestry that pointed toward the Eastern Mediterranean, toward the Middle East, toward North Africa. The Melungeons carried Rh-negative blood at rates that were elevated compared to what their mixed ancestry would straightforwardly predict. And they have been studied, argued about, dismissed, and romanticized by turns for 2 centuries without anyone arriving at a clean explanation for who they were and where they came from. The most commonly accepted current theory is that the Melungeons were formed from the mixing of free African Americans, European indentured servants, and Native Americans in the colonial period, individuals at the margins of all 3 communities who found common cause in the isolated mountain hollows when no one came to check on them. That explanation is probably substantially correct, but it does not fully account for all of the genetic anomalies that researchers have found in Melungeon communities. And it does not explain the oral traditions that some Melungeon families maintained. Traditions that spoke of ancestors who came from across the water long before the first colonial ships. Ancestors who were neither European nor African, nor the people of this land, but something else, something older. The Melungeons are not the only example. The Lumbee of North Carolina, one of the largest Native American groups east of the Mississippi, have a genetic and historical profile that does not fit the standard model of eastern woodland indigenous peoples. Their origins are disputed. Their language is gone. Their oral traditions speak of connections to other peoples, to lost colonies, to visitors and settlers from long ago. And their Rh-negative frequencies are higher than what their primarily indigenous American genetic heritage would predict. The brass ankles of South Carolina, the red bones of Louisiana and Texas, the Jackson whites of New Jersey and New York, the brown people of North Carolina. There are dozens of these mixed-race, genetically complex, historically ambiguous communities scattered across the American South and East, each carrying a genetic profile that tells a more complicated story than the standard narrative of American demographics would suggest. And in many of these communities, Rh-negative blood appears at rates that demand explanation. These communities share certain characteristics. They were isolated either by geography or by social exclusion. They maintained internal marriage practices over generations, which concentrated whatever genetic traits they carried. They existed at the margins of all official social categories, classified neither fully as white nor as black, nor as Native American under the racial taxonomies that American law has constructed and reconstructed over the centuries. And they carried genetic material that reflected the full, messy, violent, complicated reality of how individuals actually moved and mixed in colonial America in ways that the official records simply did not document. The isolation is key in population genetics. Small isolated populations are where genetic drift operates most powerfully. In a large open population, a rare genetic trait tends to be diluted over time as individuals breed with a much larger group of people who do not carry that trait. But in a small isolated population, a rare trait can become common simply through chance, simply because the founding members of that population happen to carry it, and their descendants kept marrying each other for generations. This is called the founder effect, and it is one of the most powerful forces in human genetic history. Every isolated population in America, every community that stayed in the same hollow or on the same island or behind the same mountain for multiple generations, is a natural experiment in what happens when a small founding group's genetic material gets amplified through isolation. Some of those founding groups carried elevated Rh-negative frequencies, and in their descendants that elevation was preserved and sometimes amplified while the rest of America churned and mixed and diluted and averaged out. That is another darker reason for America's unusual Rh-negative map. Not dark in the sense of sinister, but dark in the sense of hidden. Hidden in the folds of mountains, hidden in the swamps, hidden in the oral traditions of communities that the official history did not think worth recording. Let us go back to the blood itself now, because there is a physiological dimension to this story that has its own darkness. One that played out specifically in the American context in ways that cost lives for generations before anyone understood why. The problem with Rh-negative blood in medical terms is what happens when an Rh-negative woman carries an Rh-positive baby. During pregnancy, especially around the time of delivery, small amounts of fetal blood can cross the placenta into the mother's bloodstream. If the baby is Rh positive and the mother is Rh negative, her immune system encounters the Rh protein for the first time and begins producing antibodies against it. The first pregnancy is usually fine because the immune response develops slowly and the baby is typically born before the antibody levels get high enough to cause damage. But the next pregnancy is different. If the second baby is also Rh positive, those antibodies are already in the mother's bloodstream, ready and waiting. They cross the placenta. They attack the baby's red blood cells. The baby's body tries to compensate, producing more and more red blood cells to replace the ones being destroyed. In severe cases, the baby develops profound anemia. Fluid accumulates in the tissues. The liver and spleen enlarge, trying to produce enough blood to keep the organs functioning. Babies were born yellow with jaundice, swollen, struggling to breathe. Many died within hours or days. Some survived with permanent brain damage. Before 1968, when the treatment now known as Rogam was developed, there was no way to prevent this. And for American families, particularly in isolated communities where Rh-negative blood was concentrated by the founder effect, this played out as a quiet generation-spanning tragedy. Families where the mother was Rh-negative would often lose their second, third, fourth children. The first child would be fine, and then the subsequent children would come into the world already fighting for their lives. Their blood under attack by the very body that was supposed to nurture them. In the mountains of Appalachia, in the rural South, in the isolated communities where Rh-negative blood had been concentrated over generations, mothers watched their babies die and did not know why. The folk explanations were many and varied. The evil eye, a curse, God's will, something wrong with the blood, which was closer to the truth than most of the other explanations, but still did not provide any way to prevent it. The medical establishment in America for most of American history was not available to these communities anyway. The poor, the rural, the racially marginalized, the geographically isolated, these were precisely the communities where Rh-negative blood was most concentrated. And they were also precisely the communities with the least access to medical care. So the tragedy of Rh incompatibility played out most severely in the communities least equipped to understand or address it. There is a bitter irony in this. The communities that had been pushed into isolation by poverty, by racial categorization, by geographic marginalization were the communities where the founder effect had concentrated Rh-negative blood. And because of that concentration, they were also the communities most affected by Rh incompatibility, losing children generation after generation to a condition that the white medical establishment was only beginning to understand and would not solve for another century. This is another darker reason. The isolation that concentrated the blood was the same isolation that meant no one came to help when the blood turned against the children. Now, let us turn towards something that exists at the edges of this story. Something that mainstream genetics treats with polite skepticism, but that keeps surfacing in the data, in the oral traditions, and in the anomalies that researchers keep noticing and then quietly setting aside. There is a question that has been circling the study of Rh-negative blood for decades without ever quite being answered. If this trait persists at significant frequencies despite creating a reproductive disadvantage, what advantage does it provide that offsets the cost? Why has natural selection not eliminated it, or at least reduced it to the vanishingly rare frequencies that most genuinely disadvantageous traits settle into? The standard answer is that in small, isolated populations, genetic drift can maintain traits even when they are mildly disadvantageous. If a population is small enough and isolated enough, chance alone can keep a trait circulating regardless of whether it helps or hurts. And there is truth to this. It explains a lot of the Rh negative distribution story. But it does not explain everything, because some of the populations where Rh negative blood is most concentrated do not look like populations experiencing simple genetic drift. They look like populations where some kind of selection has been actively operating, where the trait has been maintained or amplified beyond what drift alone would predict. Some researchers have proposed that Rh negative blood might confer some immune advantage. That the same genetic variation that eliminates the Rh protein from the red blood cell surface might also alter the immune system in ways that provide better resistance to certain pathogens. The evidence for this is preliminary and contested, but there are a few areas where it looks interesting. Rh negative individuals appear in some studies to have different susceptibility profiles to certain parasitic infections. The Toxoplasma gondii parasite, which is carried by cats and which infects roughly a third of all humans worldwide, behaves differently in Rh negative individuals in some research findings. The behavioral and neurological effects that Toxoplasma infection appears to produce in Rh positive individuals, things like altered risk perception and subtle changes in personality, seem to be reduced or absent in Rh negative individuals. This is a very specific and very strange finding, and it has attracted a lot of attention from researchers trying to understand the parasite's relationship with human evolution. There are also some interesting findings around resistance to certain respiratory viruses. And during the COVID pandemic, several studies found weak but consistent associations between Rh negative status and reduced risk of infection or severe disease. These associations were modest. They did not survive all forms of statistical control, and they should not be overstated. But they kept appearing in study after study across different populations and different methodologies. Something was there, even if the something was smaller and more complicated than any simple immune advantage story would suggest. In the context of North America, any immune advantage from Rh negative status would have been tested and potentially selected for multiple times over the course of American history. The indigenous peoples of North America were devastated by epidemic disease after European contact. The mortality rates from smallpox, measles, influenza, and other old world diseases were catastrophic, sometimes 90% or higher in affected communities. These were genuine extinction-level events for many populations. Any genetic variant that provided even partial resistance to these diseases would have been under intense positive selection in the post-contact period. If Rh negative blood provided any resistance advantage to infectious disease, then the communities where it was concentrated, whether through the founder effect, through forced genetic mixing, or through ancient population structure that we do not fully understand, would have been the communities best positioned to survive the epidemiological catastrophe of European contact. Their descendants would carry more Rh negative blood than the broader population, because the individuals most likely to survive and reproduce were the ones carrying this variant. This is speculative. The evidence for a meaningful disease resistance advantage from Rh negative status is not strong enough to support confident conclusions. But it is the kind of speculation that becomes interesting when you lay it over the specific historical events of American colonization and notice that the populations where Rh negative blood is most concentrated today are in many cases the populations whose ancestors survived events that killed most of everyone else. The survivors carry the blood of survivors, and sometimes that blood contains traits that explain the survival. Now let us follow another thread. This one goes underground into the hidden spaces of American identity, into the communities and bloodlines that official history classified and then forgot. One of the most remarkable and least examined aspects of American demographic history is the phenomenon of racial passing. Racial passing refers to the practice by which individuals who had some degree of African, Native American, or mixed ancestry presented themselves and were accepted as white in a society where the legal and social benefits of whiteness were enormous and the penalties for being classified as non-white were severe. It has been estimated that millions of Americans of partial African American ancestry crossed the color line over the course of American history, particularly in the decades following the Civil War, when the legal system began to codify racial categories with increasing precision and the social costs of being classified as non-white reached their historical peak. These individuals did not announce what they were doing. They moved to new communities. They constructed new identities. They raised children who in many cases did not know the full story of their ancestry. They became white, at least in the social and legal sense, and they contributed their genetic material to the populations that considered themselves white. Their genetic contribution included whatever Rh-negative blood they carried. If they were descended from the complex mixed-race communities of the pre-Civil War South, they likely carried both European origin and African origin genetic material in proportions that varied from individual to individual. And the Rh-negative blood in that genetic material, whether it came from European lineages or from the African American communities where centuries of forced mixing had introduced it, went with them into the white communities they passed into. This is another mechanism by which the American Rh-negative map was shaped. Not through the obvious channels of colonial immigration or the documented movements of enslaved people, but through the hidden channel of millions of individual decisions to cross a color line that the society around them treated as absolute, but that their own biology was quietly and continuously blurring. The descendants of people who passed are everywhere in America today. Most of them do not know who their ancestors were. The genetic testing services that have become popular in the last decade have produced millions of revelations for families who thought they knew their ancestry and discovered they did not. African American ancestry in families that identified as entirely European. Native American ancestry in families that had no tribal affiliation or recognition. European ancestry in communities that had maintained strict separation from white genetic contact. The color line that American history treated as a wall was always a sieve, and the genetic consequences of that seeping are distributed throughout the American population in ways that no census, no family record, and no official history fully captures. And because Rh-negative blood follows genetic ancestry, it is distributed the same way, hidden in families that do not know why they carry it, concentrated in communities whose full genetic history has never been told. Part of an American portrait that official history drew in too few colors to show what was actually there. Let us go back now to the very ancient past, because there is one more layer to this story that belongs in the deep time before any of this history took shape. And it is in some ways the most unsettling layer of all. Around 900,000 years ago, something happened to the human family tree that geneticists are still struggling to fully understand. The evidence comes from analysis of ancient DNA and modern genomes, from the patterns of genetic variation that survive in living human populations as ghosts of events that happened nearly 1 million years in the past. What happened, as best we can reconstruct it, is this: the ancestral human population, whatever species or group of species was living in Africa at that time, experienced a catastrophic population collapse. The numbers vary depending on the analysis, but some estimates suggest that the effective breeding population of our ancestral lineage dropped to perhaps 1,000 individuals or fewer during a period that lasted somewhere between 100,000 and 300,000 years. 1,000 individuals. That is smaller than the number of people in many small towns. That is smaller than the number of people who might attend a moderately sized sporting event. That is the entire species, or the entire surviving lineage of the species, the whole thread from which all of modern humanity would eventually descend, reduced to a number small enough that you could theoretically know every person in it by name. This bottleneck, if that is what it was, would have had profound effects on human genetic diversity. When a population collapses to that size, most of the genetic variation that existed before the collapse is lost. Whatever rare traits were present in the survivors by chance become the foundation for everything that comes after. Whatever rare traits happened not to be carried by any of those 1,000 individuals disappear forever. The Rh-negative variant, or the ancestral genetic condition that eventually gave rise to the Rh-negative variant as we know it, may have been one of the traits that survived this bottleneck in some lineages and not others. If the founding population of a particular branch of humanity happened to carry this variant at elevated frequencies, their descendants would too. Regardless of whether the variant conferred any advantage, it would just be part of who they were, part of the genetic fingerprint of that particular surviving lineage. This is the deepest layer of the Rh-negative mystery in America and everywhere else. Not human history in the sense of the last 10,000 years. Human history in the sense of the last million years. The ghost of a near-extinction event that shaped the genetic possibilities of everything that came after. When you carry Rh-negative blood, you are carrying a variant that survived something. Something that nearly ended the human story before it properly began. The survival was not because of this variant. The survival was despite the enormous pressure of whatever crisis reduced our ancestors to a thread. But the variant survived along with everything else in those survivors, and it has been passing forward through the generations ever since, showing up in unexpected places, distributed unevenly across the human world in patterns that reflect the specific paths that surviving lineages took through the subsequent 1,000 centuries. In America, those paths converge from multiple directions. European colonists, enslaved Africans whose gene pool was forcibly altered by European genetic material over generations of coercion. Indigenous peoples whose origins are more complex than the standard model acknowledges. Isolated communities where the founder effect concentrated rare traits into frequencies that no one would expect. Individuals who crossed racial lines and took their genetics with them into communities that did not know they were carrying it. All of these paths converging on one continent, in one country, over 500 years of documented history, and many thousands of years of undocumented history. And the result is the Rh-negative map that researchers look at and cannot quite explain. The darker reason is not one thing. It is many things layered on top of each other. It is colonial violence. It is the forced movement of human populations. It is isolation imposed by poverty and racial categorization. It is the silence around mixed-race ancestry in a society that preferred not to see what it had created. It is the loss of indigenous history and the suppression of evidence that complicates the tidy story of who was here first. It is the reproductive tragedy that played out quietly in isolated communities for generations before medicine understood enough to help. It is the ghost of a near-extinction event nearly 1 million years old. None of these things alone explains the full picture. All of them together begin to come close. And there is one more thing, one more dimension to this story that we have not fully explored yet. It has to do with what happens to human communities under sustained trauma. With what the body remembers, with what genetics preserves, not just as a passive record of ancestry, but as an active response to the conditions that ancestral populations survived. The science of epigenetics, which studies how environmental factors can influence the way genes are expressed without changing the underlying DNA sequence, has produced some of the most disturbing and most fascinating findings in recent biology. The studies that have attracted the most attention are the ones showing that the physiological effects of severe trauma can be transmitted across generations through mechanisms that do not involve the genes themselves. The Holocaust studies are the most cited example. Research on the descendants of Holocaust survivors has found that they carry different patterns of stress hormone regulation than control populations without that ancestral history. The methylation patterns on certain genes associated with cortisol production and stress response are altered in the descendants of individuals who experienced extreme trauma, even when those descendants were born long after the trauma and never directly experienced it themselves. The body appears to have passed down something beyond the genetic sequence. It passed down a kind of physiological memory, a readiness, an altered biochemical baseline shaped by what the ancestors survived. What this means for the Rh-negative story in America is not straightforward. Epigenetics does not change blood type. You cannot acquire or lose Rh negative status through traumatic experience. The genetics of the Rh protein are not subject to epigenetic modification in that direct sense. But the distribution of Rh negative blood across populations that have experienced severe collective trauma raises a different question. Not whether trauma changed the blood, but whether the communities where Rh negative blood is most concentrated are communities whose histories were shaped by the kind of events that also shaped epigenetic patterns in their descendants. And if so, what does that convergence mean for how we understand those communities today? The African American community in the United States is the most obvious example. This is a community whose Rh negative frequencies are higher than their West African ancestry would predict because of centuries of forced genetic mixing with European populations. And this is also a community whose epigenetic research suggests carries altered stress physiology as a result of intergenerational trauma. The two findings are not the same finding, but they both point to the same underlying reality, that what happened to this community in the historical period left marks that are still present in the biological profiles of living people. The Melungeon communities of Appalachia, the Lumbee and other southeastern tribes, the isolated mountain communities of the rural South. These two are communities whose histories included sustained marginalization, violence, displacement, and the kind of multigenerational stress that epigenetic research suggests leaves biological traces. And these two are communities where Rh negative blood shows up at anomalous frequencies. This is not a causal claim. It is an observation about patterns. But patterns have a way of pointing towards something even when the mechanism is not yet understood. Now let us take a step back from the specific biology and the specific history and look at the larger picture of what this story tells us about America. America is unique among nations in the scale and the speed of the genetic mixing that created it. No other country absorbed so many people from so many different genetic backgrounds under so many different circumstances in so short a period of historical time. The process was not peaceful, and it was not voluntary, and it was not equitable. But the genetic result is a population that is in some ways more diverse and more complex than any other large national population on Earth. In most countries, elevated Rh negative frequencies reflect ancient history, the isolation of the Basque, the particular genetic legacy of the Atlantic fringe of Europe, the survival of pre-agricultural lineages in mountain refuges. These are stories that played out over thousands of years. In America, the same result, elevated Rh negative frequencies in a complex and diverse national population, was produced in 500 years, and the mechanisms were different. Not isolation and slow genetic drift, but violent displacement and forced mixing, and the founder effect operating at massive scale as enslaved populations and isolated communities concentrated unusual genetic profiles through coercion and circumstance. The rapidity is remarkable, and it means that the American Rh negative map is in a very real sense a scar. Not all of it. The European colonial contribution is straightforward and unremarkable, the simple immigration of populations that already carried the trait. But the layers beneath that straightforward story, the layers involving slavery and isolation, mixed-race communities and suppressed indigenous history, those layers are scars. They are the biological record of events that the official history of America has spent considerable energy minimizing, denying, or simply failing to examine. Blood does not lie in the way that official histories sometimes do. It carries what happened, regardless of whether anyone writes it down. And the Rh negative distribution in America is, among other things, a record of everything that official history left out. Let us spend some time in the place where this story becomes most personal, because blood is not just a statistic. It is something that moves through actual bodies, that shapes actual lives, that has consequences for actual families. The Rh incompatibility problem that we discussed earlier, The way an Rh-negative mother's immune system can attack an Rh-positive baby in subsequent pregnancies was a quiet catastrophe for many American families before modern medicine understood and addressed it. The treatment, Rogum, was developed in 1968. Before 1968, there was nothing to be done. The condition was recognized. The mechanism was understood from the 1940s onward. But understanding without treatment is a particular kind of cruelty. In the years between when the mechanism was understood and when the treatment was developed, doctors could watch what was happening in families with Rh incompatibility, could explain why the second or third child was dying, could offer the grim comfort of scientific explanation alongside the inability to intervene. For the families affected, the period between 1940 and 1968 was one where they had the added burden of knowing what was killing their children without having any way to prevent it. And the families most affected were precisely those communities where Rh-negative blood had been concentrated by history. The mountain communities of Appalachia, the mixed-race communities of the rural South. The populations descended from the genetic complexity of the colonial and antebellum periods. These communities, already marginalized from the mainstream of American medical care, were also the communities where the founder effect had elevated Rh-negative frequencies and where therefore the incidence of Rh incompatibility was highest. The same marginalization that excluded these communities from access to adequate health care was also through the mechanisms of isolation and founder effect, creating a health burden within those communities that adequate health care would have been required to address. It is a vicious circularity, and it is one of the most concrete and specific ways in which the historical forces that shaped the American Rh-negative map also caused harm to the people whose blood carries that history. Access to Rogum changed this. After 1968, Rh incompatibility became a manageable condition rather than a recurring family tragedy. An Rh-negative woman who receives appropriate prenatal care can have as many healthy Rh-positive babies as she wants. The injection of Rh immune globulin after delivery prevents the sensitization that would otherwise trigger the immune attack on subsequent pregnancies. The treatment is simple, effective, and almost universally available in developed healthcare systems. But in America, access to prenatal care has never been universal. And the communities where Rh-negative blood is most concentrated, the poor communities, the rural communities, the racially marginalized communities, are also the communities with the least reliable access to prenatal care. So while Rogum theoretically solved the Rh incompatibility problem in 1968, the problem continued to play out in the communities least connected to the healthcare system for decades afterward. Infant mortality data from rural Appalachia and from African American communities in the South continued to show the signature of Rh incompatibility. Babies dying of hemolytic disease of the newborn in years when the condition was theoretically preventable, because the prevention required access that these communities did not always have. The biological legacy of historical trauma concentrated in these communities through the mechanisms we have described was still causing harm long after the medical solution had been found. That is one of the darkest dimensions of this story. Not that the blood itself is dark, but that the history that shaped its distribution also shaped the conditions that made it dangerous. And the conditions that made it dangerous were precisely the ones that the same historical forces created. Poverty, marginalization, exclusion from the mainstream of American life and American medicine. Let us now go somewhere different. Let us step outside the documented history and into the space where evidence thins out and the story becomes something closer to mystery. There is a category of evidence in American archaeology that researchers sometimes call out-of-place artifacts. These are objects found in American archaeological contexts that appear to have origins inconsistent with the standard model of American prehistory. Roman coins found in Kentucky. Viking rune stones in Minnesota. Celtic Ogham inscriptions carved into rocks in the American Southwest. An ancient Hebrew text found in Ohio. Bronze Age tools that match Mediterranean typology more closely than anything from the Asian traditions that standard archaeology expects to find in pre-Columbian North America. The academic establishment's response to out-of-place artifacts is generally dismissal. These objects are usually explained as hoaxes, as later depositions, as misidentifications, and in many cases that dismissal is entirely warranted. The history of American archaeology is littered with forgeries, with wishful thinking, with the imposition of preconceived narratives onto ambiguous evidence. The Kensington runestone, which a Minnesota farmer claimed to have found in 1898, purports to be a Norse inscription from 1362 and has been the subject of 120 years of debate without resolution. Most professional linguists and archaeologists consider it a 19th century hoax. Some do not, but the wholesale dismissal of all anomalous evidence does its own kind of damage. It creates a model of American prehistory that is artificially clean, artificially simple, artificially consistent with a narrative that serves certain political purposes. The narrative that North America before Columbus was a continent populated exclusively by the descendants of Bering Land Bridge migrants, living in ways that did not include significant contact with the rest of the world, is a narrative that has political uses. It simplifies the question of indigenous sovereignty. It eliminates the complications that arise when the pre-Columbian story turns out to be as messy and layered as every other period of human history. And the genetic evidence increasingly suggests that the clean narrative is wrong. Even if the specific claimed examples of pre-Columbian contact, the runestones, the coins, the inscriptions, are individually suspect, the genetic evidence does not require us to accept any particular artifact claim. It just requires us to accept that the American population before 1492 was not genetically homogeneous in the way the standard model predicts, and that it included elements that point toward contact histories that the standard model does not accommodate. The X haplogroup is one of the most discussed examples. Haplogroups are groupings of genetic variants that trace specific ancestral lineages. Most Native American populations belong to haplogroups that trace straightforwardly to Northeast Asian origins. This is exactly what the Bering Land Bridge model predicts, and the genetic evidence for it is strong. But a small percentage of Native Americans, distributed in a pattern that is geographically concentrated in the eastern United States, particularly among the Ojibwe and Algonquian peoples of the Great Lakes region, belong to haplogroup X. And haplogroup X is found in Europe and in the Middle East at significant frequencies, but is essentially absent from Northeast Asia, the expected source of all ancestral Native American populations under the standard model. How did haplogroup X get to the eastern Great Lakes without passing through Northeast Asia? The standard explanations involve the loss of intermediate populations in Asia through genetic drift, or the identification of rare X carriers in Asian populations that might have contributed to American founding populations without leaving other detectable traces. These explanations are possible, but they are not satisfying, and they rely on a convergence of improbable circumstances. The alternative explanation is that some ancestral population reached North America through an Atlantic route, bringing haplogroup X from its western Eurasian range into the eastern part of the continent. This population would have been genetically distinct from the Northeast Asian-derived populations that came through the Bering Land Bridge. They would have contributed a genetic signature that shows up in eastern North American populations, but not in western ones, which is exactly the distribution pattern that we observe. And if that Atlantic route population came from the zone where Rh-negative blood is most concentrated, from the Atlantic fringe of Europe or North Africa, they may also have brought Rh-negative blood with them. This would provide a mechanism for anomalous Rh-negative frequencies in certain eastern North American populations that does not require post-Columbian European contact. It would push the introduction of Rh-negative blood into the American genetic story back by potentially thousands of years, into a period before Columbus, before the Norse, before any of the documented contacts between the Old World and the New. This is speculation built on speculation. The case for Atlantic route pre-Columbian migration to North America is suggestive but not proven. The connection between haplogroup X distribution and Rh negative distribution is a hypothesis, not a finding, but it is a hypothesis that is consistent with the data we have. And it is a hypothesis that would explain anomalies that the standard model does not explain. And in the space between what we can prove and what the evidence suggests, there is a story. A story about a people who crossed an ocean before history recorded them, who arrived in a land already inhabited, who contributed their genetic material to that land's populations in ways that left echoes in the blood of their descendants thousands of years later. Echoes that show up as elevated Rh negative frequencies in communities that the standard story of America does not fully account for. Let us sit with that for a moment, because the practice of sitting with uncertainty, of living in the space between what is known and what is merely suspected, is one of the oldest human practices there is. Long before we had genetics, long before we had archaeology, long before we had written history of any kind, people sat with the mystery of their own origins. They told stories about it around fires in the dark, trying to make sense of who they were and where they came from and why the world was the way it was. We are doing the same thing with different tools. The tools are more precise. The data is more extensive. But the fundamental activity, the reaching into the dark past to find the shape of something that cannot be directly seen, is the same activity that human beings have always engaged in. And the humility required to do it honestly, to say I am not certain, to say the evidence points here, but does not prove, to say there is more that we do not know than we do know. That humility is also something that human beings have always practiced in their better moments. The American Rh negative story is a mystery that does not have a clean resolution. It has layers. It has documented history and suppressed history and contested history and genuinely unknown history. It has genetics that tell us things that the official record does not. And it has gaps in the genetics that leave room for possibilities that we have not fully imagined yet. And it has the blood itself, moving through millions of American bodies, carrying this ancient absence of a protein. This deletion that survived a near-extinction event nearly 1 million years ago, that traveled with human populations across continents and oceans and centuries, that accumulated in America through the specific, violent, complicated, and largely untold mechanisms of American history. Now let us explore something that might seem unrelated, but is actually at the heart of why this question matters. The relationship between what we know about our bodies and what we know about our histories. In most countries, if you want to understand the distribution of a genetic trait, you can trace it through documented populations. You have records, you have genealogies, you have census data and migration records and family trees maintained across generations. You can look at a genetic map and match it against a historical map, and the patterns align in ways that make sense. In America, this process breaks down in significant ways. The historical record for large segments of the American population is fragmentary, or deliberately destroyed, or simply never created. Enslaved people were not allowed to maintain family records. Indigenous communities saw their records destroyed, their oral traditions suppressed, their continuity as communities forcibly interrupted by removal and assimilation policies. Mixed-race communities existed in the legal margins, classified inconsistently, recorded inconsistently, sometimes not recorded at all. People who crossed racial lines to pass as white destroyed whatever evidence might have connected them to their actual ancestry. The result is a national population with a genetic profile that the historical record cannot fully explain. When geneticists look at the American genome, they see things that do not match the documented story. They see African ancestry in populations that identified as entirely white. They see European ancestry in populations that maintain strict separation from European contact. They see Native American ancestry in communities with no tribal affiliation. They see genetic markers that point to origins not accounted for in any family record or census category. The blood remembers what the records forgot. or what the records were designed to obscure. This is not a minor methodological puzzle for geneticists. It is a profound statement about the relationship between official history and biological reality in America. The country was built in part on the fiction that its populations were categorically distinct, that the color line was real and meaningful and could be maintained, that ancestry was knowable and controllable. The genetics say something very different. The genetics say that this was never true, that the mixing was always happening, that the categories were always fictions imposed on a biological reality that did not honor them. The Rh negative distribution is one thread in that larger genetic story. It is one of the signals that persists in the American genome to tell a more complicated truth than the official narrative allows. And because Rh negative blood is a visible, testable, medically significant trait, it is one of the clearest examples of how the hidden complexity of American genetic history has concrete lived consequences for people in the present. But before the final rest arrives, let us spend more time in some of the places this story has opened that deserve deeper exploration. Because a mystery this old and this layered does not yield its full depth in one pass. It opens like a landscape reveals itself from altitude. First the broad shapes, then the particular details, then the fine textures that only become visible when you slow down and look carefully. There is a dimension of the American Rh negative story that we have touched on but not fully examined. It has to do with blood supply and emergency medicine, and it is a dimension where the abstract history becomes immediately and concretely present in the lives of people alive right now. Every year in the United States, the blood banking system faces a recurring crisis. There is never enough O-negative blood. O-negative is the universal donor type for red blood cells, the blood that emergency rooms reach for when there is no time to type and cross-match a patient. When someone is bleeding out, and every second without compatible blood brings them closer to death. Trauma victims, mothers hemorrhaging during childbirth, children in accidents, surgical patients whose operations have unexpected complications. All of these situations demand blood immediately, before the laboratory can determine what the patient's own blood type is. And the blood that meets that demand is O-negative. But the people who can donate O-negative blood are only about 8% of the American population. And because whole blood cannot be stored indefinitely, because the red cells in donated blood degrade over time, there is a constant need for fresh donations from that 8%. The blood banking system is always operating with less O-negative supply than the demand would prefer. This shortage is not unique to America. It is a global phenomenon wherever blood banking systems operate. But in America, the specific history we have traced has shaped the O-negative supply situation in specific ways. The communities where O-negative blood is most concentrated because of the founder effect and the historical mixing patterns we have described are often the communities that have the most complicated relationships with the healthcare and blood banking systems. Mountain communities with limited access, rural communities with fewer donation sites, communities whose historical experiences with medical institutions have given them legitimate reasons for caution about participation. This creates a situation where the blood that the emergency medicine system most needs is concentrated in the communities that the system has most historically failed and most rarely reaches. It is another circularity in the darker pattern. The marginalization that concentrated the blood is also the marginalization that makes the blood harder to collect. Blood banking organizations in America have recognized this and have made efforts to expand donation programs into underserved communities. Mobile donation units, community partnerships, educational campaigns designed for specific communities. These efforts have had real effects, but the underlying pattern, the one rooted in the history we have traced, persists. And there is something worth sitting with here. The O-negative blood that saves lives in American emergency rooms, blood that saves the lives of people from every background and every community, is disproportionately produced by communities that have historically been marginalized and excluded and exploited by the broader American society. The descendants of enslaved people, the mountain communities whose isolation reflects their historical exclusion from the American mainstream, the mixed-race communities that existed in the legal margins for generations. These communities give the blood that keeps the medical system functioning for everyone, including with some frequency the people whose ancestors were responsible for the historical circumstances that concentrated this blood in these communities in the first place. That is a particular kind of dark irony, not an unusual one in the history of how marginalized communities have contributed to the societies that marginalized them, but worth naming, worth being clear about, because the clarity matters. The blood that saves lives does not carry its history with it into the emergency room. It does not announce where it came from or what produced it. It just works, doing what blood does, keeping bodies alive. But the history of where it came from is real, and the pattern of who donates it, and who needs it, and how the two connect is part of the same complicated story we have been tracing all night. Let us go to another specific story, one that illustrates the intersection of blood type, medical history, and American historical complexity in a very particular and very human way. In the years immediately following the Second World War, American medicine went through a period of rapid expansion and systematization. Blood banking, which had been developed and refined during the war to manage the enormous demands of battlefield medicine, was being adapted to civilian healthcare. Blood typing was becoming routine. The Rh factor, discovered in 1940, was being incorporated into the medical understanding of blood compatibility. And doctors were beginning to understand that some of the infant deaths they had been seeing for generations were not just bad luck or divine will, but the product of a specific biological mechanism that could in principle be managed in the hospitals of the rural South, in the clinics that served Appalachian communities, in the medical facilities that reached, however inadequately, into the isolated communities where Rh-negative blood was concentrated. This new knowledge arrived in a specific and often heartbreaking form. Doctors began being able to tell families why their second or third or fourth child had been born sick and dying. They could explain the mechanism, but explaining the mechanism was not the same as being able to fix it. The treatment, Rogum, would not arrive until 1968. So for the 2 decades between when the mechanism was understood and when the prevention became available, doctors in these communities experienced a particularly painful form of knowledge. The knowledge of what was happening and the inability to stop it. There are accounts in the medical literature from this period that are worth sitting with. A doctor in rural Tennessee, writing about a family he had treated where the mother had lost her third, fourth, and fifth children to hemolytic disease of the newborn. The first child, the one born before the mother's immune system was sensitized, was healthy. The subsequent children were not. The doctor could tell the family what was happening. He could not stop it from happening. He could only watch and document and wait for the science to catch up to the clinical reality. That period of wait between understanding and solution was longer for these communities than for communities with better access to research institutions and medical centers. The developments that would eventually produce Rogum were happening in academic medical centers in New York and Liverpool and a handful of other places. The information traveled slowly to rural practices and isolated communities. And during the years that it traveled, babies continued to die. The families that went through this carry it in their histories, even when they do not know the clinical explanation for what happened. The old people who remember losing siblings, the family trees with the gaps where children died young, the oral traditions that speak of something wrong with the blood, something that came for the children. These memories are not abstract. They are specific, and they belong to specific people in specific places. And they are part of the human dimension of the same story that we have been tracing through genetics and history and deep time all night. Now let us spend some time with something that is genuinely puzzling and that I want to be honest about the limits of my knowledge concerning. It has to do with the relationship between Rh-negative blood and what researchers sometimes call population stratification. Population stratification is the technical term for the fact that human populations are not randomly mixed. They have structure. Certain genetic variants cluster together in certain communities because the people in those communities share common ancestry and have been reproducing within that community rather than randomly across the entire human population. This structure has implications for medical research because it can create spurious correlations between genetic traits that appear linked simply because they co-occur in the same structured population. For the Rh-negative story, this means that some of the reported associations between Rh-negative status and various health outcomes or personality traits or disease susceptibilities may be artifacts of population stratification rather than direct effects of the Rh factor itself. If Rh-negative people in a particular study happen to share other genetic traits because they are disproportionately drawn from a particular ancestral population where those traits are common, then any association between Rh-negative status and those outcomes might reflect those shared traits rather than the Rh factor specifically. This is a genuine methodological challenge for research in this area, and it has contributed to the inconsistency of findings about Rh-negative health effects that we noted earlier in this story. Different studies drawing from different populations with different patterns of population structure find different associations. Not necessarily because the true effect of Rh-negative status varies between populations, but because the other genetic variants that co-occur with Rh-negative status in those populations are different. In the American context, this is particularly complicated because the mechanisms we have described, the founder effect, the forced genetic mixing, the racial passing, all produce patterns of population structure that are unusually complex and unusually different from what most genetic research assumes. Research conducted primarily in populations of European ancestry finds one set of associations. Research conducted in populations with more complex ancestry finds different patterns. And the communities where Rh-negative blood is most concentrated in America, because of the specific historical circumstances we have described, often have ancestry patterns that do not fit the standard models that genetics research was built around. This is not just a methodological puzzle for researchers. It is another way in which the specific history of how American blood came to be distributed the way it is has concrete consequences for how we understand that blood today. The research tools we use to study genetic effects were developed with certain assumptions about population structure. Those assumptions fit some populations and do not fit others. The populations where American Rh-negative blood is most concentrated are often the ones where the assumptions fit least well. And so the medical and scientific understanding of what Rh-negative blood means in terms of health effects and disease associations and the various claims that circulate about it is built on a foundation that is shakier than most people realize, because the foundational research was done in populations that are not representative of the full range of populations where this trait appears in America. This is another reason for humility, not just about the historical claims and the speculative archaeology, but about the medical and scientific claims as well. The story of Rh-negative blood in America is one where almost every level of the inquiry reveals more complexity than the previous level suggested, where every answer generates more questions, where the closer you look, the more uncertain things become rather than more clear. That is appropriate. That is what honest engagement with complexity looks like. It is not a comfortable place to be, but it is a more honest place than the comfortable simplicity of explanations that do not actually fit the evidence. Let us now go to a very specific place, a specific community in a specific part of America that illustrates everything we have been discussing in the most concentrated possible form. The mountains of western North Carolina, eastern Tennessee, southwest Virginia, and eastern Kentucky form one of the most genetically isolated regions in the United States. The people who settled these mountains in the 18th and early 19th centuries found themselves in terrain that made subsequent migration both into and out of the region difficult. The hollows were deep, the ridge lines were high, the roads for most of American history were either poor or non-existent. Communities formed in particular hollows and stayed there. Families intermarried over generations. The genetic mixing that characterized most of the American population, mixing through mobility and urbanization and the constant churning of the frontier, did not happen here in the same way. The result is a set of communities that are genetic time capsules. Researchers who have studied the genetics of Appalachian populations have found that they maintain higher genetic diversity in some respects than other American populations because the isolation preserved variants that would have been diluted in more mobile gene pools, but lower diversity in other respects because the small founding populations and high rates of local endogamy produced the classic signature of the founder effect. Among the traits that were concentrated in certain Appalachian communities through this process was Rh-negative blood. In some of the most isolated hollows, Rh-negative frequencies rose above 20%, and in some family clusters approached 30% or higher, levels comparable to Basque Country and among the highest in any American community. This concentration had consequences. The Rh incompatibility problem, which we discussed at length earlier, played out with particular severity in these communities. In a community where Rh-negative frequency approaches 30%, the probability that an Rh-negative woman will have an Rh-positive baby, triggering the incompatibility response in subsequent pregnancies, is high. And in a community where the same family is intermarried generation after generation, the pattern could recur in the same family multiple times. A woman might lose a second child, then a third, then a fourth, without ever having access to the medical explanation or the medical solution. The folk memory of these losses is preserved in Appalachian culture in ways that outside observers have sometimes misread. The tradition of burying babies near the home rather than in churchyards, which was common in some Appalachian communities, reflects in part the frequency of infant death in these communities. The particular kinds of grief that Appalachian culture developed around infant loss, the specific mourning traditions, the particular way that bereaved parents were integrated into community life. These things reflect a frequency of infant death that was shaped in part by the genetics of the community, by the concentration of Rh-negative blood in a founding population whose descendants continued to marry primarily each other. And yet, the connection was invisible. The community knew that babies died. They did not know why. They developed explanations from their own cultural resources, from their religious traditions, from folk medicine that was sometimes surprisingly sophisticated in other respects, but had no framework for understanding what was happening with the blood. The women in these communities who survived and produced large families despite losing children to Rh incompatibility were in a demographic sense selected for resilience. They were the ones who kept trying despite grief. Their descendants, the surviving communities of modern Appalachia, carry both the genetic legacy of the founding populations and the cultural legacy of the particular kind of endurance that was required to maintain community continuity through the losses that genetic circumstance imposed. This is not a story about failure. It is a story about survival, about the human capacity to persist through loss and confusion and circumstances that are never explained and never understood. The people in these mountain communities did not know what their blood was doing to their children. They just kept going because that is what people do. And the genetic and cultural patterns that resulted from that persistence are part of what makes American Appalachian communities distinct. Part of what marks them as communities shaped by specific pressures that most of America has never experienced in the same concentrated form. Now let us turn to another thread, one that takes us from the mountains of Appalachia to the coast, and then across the ocean to the very ancient place where this whole story may have begun. The Solutrean hypothesis is one of the most contested ideas in American archaeology. Developed most prominently by archaeologist Dennis Stanford and his collaborators over the past few decades. It proposes that some of the earliest human migrations to North America came not from Asia through the Bering Land Bridge, but from Europe, following the edge of the North Atlantic ice sheet during the last glacial maximum approximately 20,000 years ago. The ice edge would have extended far into the Atlantic, creating a continuous frozen surface along which people with ice-adapted survival skills, people who lived and traveled on ice, could theoretically have traveled from Western Europe to the eastern coast of North America. The tool technology argument for the Solutrean hypothesis is based on similarities between the Solutrean tool tradition of Western Europe and the Clovis tradition of North America. Both traditions produce distinctive fluted stone points using similar techniques. The argument is that this technical similarity is too specific and too detailed to be coincidental. Critics of the hypothesis point out that the distance between Solutrean Europe and Clovis North America is enormous, that the ice edge travel route is speculative, that the tool similarities might reflect independent parallel development rather than direct cultural connection, and that genetic analysis of ancient American populations has not found the clear Western European genetic signatures that the Solutrean hypothesis would predict. The last point is important. If significant populations of Western Europeans had reached North America 20,000 years ago, we would expect their genetic contribution to show up in the ancestral genetic profiles of Native American peoples, especially those in the eastern United States. And the genetic evidence for a clear Western European founding contribution to Native American ancestry is not there, at least not at the level that a major founding migration would produce. But this does not necessarily disprove the Solutrean hypothesis. It might simply mean that the Solutrean migrants were not numerous enough to leave a strong genetic signal in the gene pool of a continent that was subsequently populated largely by the much larger wave of Bering Land Bridge migrants. A small founding population arriving early and being largely absorbed by or replaced by later more numerous migrants would leave only faint genetic traces. Those traces might be findable if you knew exactly where to look, but they might be too faint to register clearly in studies designed to characterize general population ancestry. The haplogroup X distribution that we discussed earlier is potentially one of those faint traces. There are others that have been proposed, other anomalous genetic signals in Eastern North American populations that point toward Western Eurasian origins rather than Northeast Asian ones. None of them are conclusive. Together, they suggest something was happening that the standard model does not fully capture. And if Solutrean or pre-Solutrean migrations from Western Europe or North Africa did occur, they would have come from precisely the populations where Rh-negative blood is most concentrated. The Atlantic fringe populations of Western Europe, the North African coastal populations where Rh-negative frequencies are anomalously high relative to surrounding regions. These are the populations that a North Atlantic ice age migration route would draw from. These are the populations whose descendants include the highest Rh-negative frequencies in the Old World. Which means that if the Solutrean hypothesis is correct, or even partially correct, North America may have received its first significant Rh-negative genetic contribution not from Columbus and the colonial-era Europeans, but from hunters following the ice edge 20,000 years ago. From people whose names we will never know, whose languages vanished without record, whose tools were eventually replaced by other traditions, but whose blood may still be detectable as a faint signal in certain American populations that carry anomalously high Rh-negative frequencies. This is the deep time layer of the American Rh-negative story. Ancient beyond any recorded history. Ancient even beyond the oral traditions that might preserve some memory of events this remote. Visible only in the genetics, and they're only as a whisper, a trace, something that might be there or might be the overlap of other signals that we cannot yet disentangle. The further back you go, the less certain everything becomes. That is always the case with deep history. The evidence thins, the interpretations multiply. The story becomes more a matter of hypothesis and inference than of documented fact. But the thinning of evidence does not mean the story is not there. It just means that we are reading it through a medium that preserves only fragments, and that we must hold our conclusions lightly, with awareness of what we do not know. What we do know is that the American Rh negative map has multiple explanations operating at multiple timescales. The most recent explanations, the colonial-era European immigration, the forced genetic mixing of the plantation system, the founder effect in isolated communities, the racial passing that distributed genetic material across official racial boundaries. These are all documented and reasonably well understood. They are darker in various ways for various reasons. They reflect the specific and often violent history of how America was populated and how its populations were controlled and mixed and categorized over the last 5 centuries. The older explanations, the possible pre-Columbian Atlantic migrations, the anomalous genetic signals in certain indigenous communities, the deep time questions about bottlenecks and founding populations, and the paths that surviving human lineages took through the last million years. These are darker in a different sense. Darker because they are less visible. Darker because the evidence for them is fragmentary and contested. Darker because they involve the loss of entire peoples and their stories, the disappearance of whatever languages and traditions and memories they carried, leaving only the faint genetic traces in their descendants' blood as evidence that they were ever here. All of these layers contribute to why America carries more Rh negative blood than almost any other country. And all of them carry their own kind of weight, their own kind of darkness. The darkness of violence done and remembered in the genome. The darkness of things lost and not recoverable. The darkness of official history's silence about what actually happened. And the darkness of deep time, of events so old that they can only be glimpsed in the patterns of molecular variation in living cells. Events that shape the world we live in without leaving any other trace than the blood. Let us spend some time now with the people who are in the middle of all of this. The actual human beings who carry Rh negative blood in America today, who live with its medical implications, who may or may not know the complicated history that shaped the genetic inheritance they carry in every cell of their bodies. There is something singular about finding out your blood type. For most people, it is a piece of medical administrative information, something noted on a card or in a record, useful in emergencies and for pregnant women and for anyone who needs a transfusion. It does not for most people carry much emotional weight beyond the practical. But for some people, learning they are Rh negative opens a door into a kind of questioning that goes much further than the medical. There are online communities, millions of members strong, built around the experience of being Rh negative. Some of these communities are focused on practical medical matters. Some have drifted into the more speculative territory of Rh negative exceptionalism. The idea that Rh negative blood marks its carriers as somehow different from other humans in ways that go beyond the presence or absence of one protein. The ideas in these communities range from the medically interesting to the historically curious to the genuinely bizarre, and they have been circulating on the internet with increasing intensity for the last 15 years. What draws people to these communities is worth understanding, not to endorse the more extreme theories they propagate, but because the emotional reality behind the inquiry is genuine. People who find out they are Rh negative often feel that this piece of information connects to something they have always sensed about themselves. A sense of being different, a sense of carrying something that other people do not carry. A curiosity about ancestry and origin that goes beyond what the standard family history can satisfy. For Americans specifically, this curiosity is often intensified by the specific conditions of American identity. In a country where ancestry is complicated by migration, by forced displacement, by racial categorization systems that obscured actual genetic history, many Americans genuinely do not know who they come from in any deep sense. The family record stopped somewhere. The stories were not passed down. The genetic reality was different from the official story. And when someone who feels this disconnection from ancestry discovers that their blood carries a trait that marks them as part of an unusual global minority, the desire to know what that trait means and where it came from is entirely understandable. The problem is that the internet communities built around Rh-negative identity often provide answers that are more satisfying than they are accurate. The ancient alien narratives, the royal bloodline theories, the idea that Rh-negative people are the descendants of a pre-flood civilization or a separate human species or a divine intervention in human genetics. These narratives are compelling because they are large and dramatic, and because they honor the sense of distinction that Rh-negative people bring to the inquiry, but they are not supported by the evidence, and more importantly, they are not actually the most interesting story. The most interesting story is the true one. And the true story is stranger and darker and more human than any of the mythological alternatives. It involves real people in real historical circumstances making decisions and being subject to forces that left marks in their blood that their descendants carry today. It involves the specific violence of American history operating at the level of DNA, producing a genetic landscape that reflects everything the official story leaves out. That is a more disturbing story than Ancient Aliens, but it is also a more meaningful one because it is about actual human suffering and actual human resilience. Because it connects living people to ancestors whose stories were suppressed or forgotten. Because it says something true about the relationship between history and biology, about the way that what happened in the past lives on in the bodies of the present. The people who carry Rh-negative blood in America today are not a special category of human being. They are not the descendants of extraterrestrials or the survivors of a lost civilization in any mystical sense. But they are in a very real and traceable sense the descendants of people whose histories were complicated and often painful, whose genetic material was moved and mixed and isolated and concentrated by forces they did not control, and whose blood preserves the record of all of that in a form that modern science is only beginning to fully read. That is worth knowing, not because it makes Rh-negative people superior or different in any meaningful behavioral or cognitive sense, but because knowing it connects living people to actual history, to actual ancestors, to the actual complicated reality of what it meant to survive in America across the centuries during which this country was being built. Let us now examine one more layer, one that connects the American story to something much larger, to the global picture of Rh-negative distribution and what it tells us about human history at the species level. The global distribution of Rh-negative blood is not random. We have established this. It clusters in the Atlantic fringe of Europe and in isolated populations elsewhere. It is rare in Asia and Africa, and in indigenous American populations. And in America, it is elevated for the specific historical reasons we have been exploring. But when you stand back from all of this and look at the total pattern, something interesting emerges. The highest concentrations of Rh-negative blood in the world are concentrated along a particular band of latitude in the Northern Hemisphere. In the regions that were most affected by the climate changes of the last ice age, the populations with the highest Rh-negative frequencies, the Basque, the Irish, the Welsh, the Highland Scots, the Berbers of North Africa, certain highland populations in the Caucasus and Central Asia, all live in geographic areas that were either refugia during the last glacial maximum or were rapidly colonized by populations from those refugia as the ice retreated. They are the populations that survived in the places where human beings could survive during the deepest freeze of the last ice age, and then spread out from those survival zones as conditions improved. This pattern suggests something about the origin and distribution of Rh-negative blood that goes beyond the simple story of genetic drift in isolated populations. It suggests that there may be an association between Rh-negative blood and the specific selective pressures of ice age survival in the North Atlantic region. Something about the conditions of that region during that period may have maintained or amplified Rh-negative frequencies in the populations that lived through it. What those conditions might have been is not clear. Cold climate adaptation, dietary specialization, resistance to pathogens that were prevalent in ice age refugia but not elsewhere. The answer is not known, but the geographic pattern is real, and it suggests that Rh-negative blood has an Ice Age signature, that it was maintained and possibly selected for in specific ways in the northern Atlantic zone during the period when that zone was an environmental extreme. And America, settled disproportionately by the descendants of exactly those Atlantic fringe populations, carries that Ice Age signature in its genetic profile. The Rh-negative blood that shows up in America above the global average is in large part the genetic legacy of populations that survived specific conditions that applied in specific places during a specific period of deep Earth history. The people who came to America brought that legacy with them, and the specific circumstances of American history, as we have explored at length, amplified it in certain communities through the mechanisms of isolation and forced genetic mixing and founder effect. The full picture, from the Ice Age refugia of the North Atlantic to the colonial plantations of the American South, from the isolated mountain hollows of Appalachia to the mixed-race communities of the eastern coastal plain, from the pre-Columbian mysteries of certain anomalous indigenous genetic signals to the racial passing of millions of Americans crossing the color line in the 19th and 20th centuries. The full picture is a story that spans 100,000 years and more, that touches every darker corner of American history, and that is written in the blood of millions of people who live in America today. Most of them do not know this story. Most of them carry their Rh-negative blood as an administrative fact, something noted in a medical record and rarely thought about. But the story is there in the genetics, waiting to be read. And now, sitting in the dark listening to this, maybe you are one of the people who carry it. Or maybe you know someone who does. Or maybe you are here for the same reason that human beings have always gathered around fires and listened to stories about where they came from. Not because the specific details apply to you, but because the question of where we come from and what it means is one of the fundamental questions of human consciousness. One that does not have a final answer, but that is worth asking in as much depth and with as much honesty as we can bring to it. The blood carries the history, and the history is darker and stranger and more human than the simple story of European immigration that textbooks prefer. That is the truth behind why America has more Rh-negative blood than almost any other country. It is not one truth. It is many truths layered on top of each other, each one reflecting a different chapter of the complicated story of how this country came to be, what it is, and who it came to contain within its borders. There is a category of knowledge that falls between the documented and the speculative, between the proven and the merely suggested. It is the space where most of the interesting questions live, the space where the evidence points towards something but does not prove it, where the pattern is visible but the mechanism is uncertain, where the data asks questions that the current tools cannot fully answer. This story has operated in that space throughout. And one of the places where it enters that space most deeply is in the question of what, if anything, the concentration of Rh-negative blood in certain American communities means for those communities today. Not in a medical sense, though the medical implications are real, and we have discussed them, but in the deeper sense of whether carrying a particular genetic heritage from a particular complicated history has any relationship to how people experience the world, to who they are, to what their communities have developed as cultures of survival and resilience in response to conditions that the rest of America did not face in the same concentrated form. This is delicate territory. The history of using genetic differences to make claims about behavioral, cognitive, or cultural differences between human groups is one of the ugliest chapters in the history of science. It has been used to justify slavery, colonialism, genocide, and every form of discrimination that has ever been visited on one group of human beings by another. The correct scientific consensus, supported by overwhelming evidence, is that there are no meaningful cognitive or behavioral differences between human populations that can be attributed to genetic variation. The human brain in its fundamental capacities is the same across all populations and all ancestry groups. The differences that exist between individuals and communities are the product of culture, history, circumstance, and experience, not of genes. Nothing in the Rh-negative story contradicts that consensus. Rh-negative blood does not make people smarter, more intuitive, more creative, more spiritual, or more capable in any dimension than Rh-positive blood. The claims to that effect, which circulate widely in online Rh-negative communities, are not supported by evidence and should not be taken seriously as scientific propositions. But there is something else worth considering. The communities where Rh-negative blood is most concentrated in America are in many cases communities that develop specific cultural responses to specific historical pressures. The Appalachian communities developed a culture of self-reliance, of maintaining traditions despite isolation, of preserving ways of life that the broader American mainstream has often dismissed as backward or provincial, but that represent sophisticated adaptations to a particular environmental and social reality. The African American community developed one of the most extraordinary cultures of resilience and creativity in the history of the world, transforming the experience of traumatic displacement and sustained oppression into music and art and literature and religious practice that has shaped global culture in ways disproportionate to the community's size. The mixed-race communities of the American Southeast developed complex strategies for navigating a social system that tried to force them into categories that did not fit their actual experience, developing in the process a kind of cultural ambidexterity, an ability to exist in multiple worlds simultaneously. That is its own form of sophistication. These cultural developments are not the product of the genetics of Rh-negative blood. They are the product of the historical circumstances that shaped these communities. But those historical circumstances are the same circumstances that shaped the genetic distribution of Rh-negative blood in these communities. The isolation that concentrated the blood was also the isolation that shaped the culture. The forced mixing that elevated Rh-negative frequencies in the African American gene pool was also the forced mixing that contributed to the cultural syncretism that produced so much of what is most distinctive about African American culture. The marginalization that excluded these communities from the mainstream of American life was also the marginalization that forced these communities to develop internal resources of great depth and variety. So the genetics and the culture are not causally connected in the sense of one producing the other, but they are both products of the same historical forces. They are both aspects of the same story, and understanding the genetics, understanding why the blood is distributed the way it is, illuminates the history in a way that understanding the culture alone does not, and vice versa. This is why the story of American Rh-negative blood matters beyond the purely medical or the purely genealogical. It is a different way of reading American history, a way of reading it through the body rather than through the documents, a way of finding the things that documents were designed to obscure, because the body does not obey the same silences that official history demands. Let us look now at what happens when communities discover this history. When individuals or communities learn the full complexity of their genetic ancestry, when the neat categories that official history provided turn out to be more complicated than anyone admitted, when the blood reveals what the records concealed. The explosion of direct-to-consumer genetic testing over the last 15 years has produced millions of these revelations. People who grew up with one story about their ancestry have received genetic reports that told a different story. The family that considered itself entirely of English heritage discovered 4% West African ancestry. The person who identified as Native American on the basis of family oral tradition discovered minimal indigenous ancestry, but substantial European and African ancestry. The person who considered their family entirely white for generations discovered that their great-great-grandparent had crossed the color line, and that their family had been living inside a fiction ever since. These revelations are not comfortable for everyone. Some people find them liberating, a release from categories that never quite fit. Others find them destabilizing, threatening to the sense of identity and community that the previous story provided. And in America, where racial identity has enormous social, legal, and political significance, The revelation that your genetic ancestry does not match your social identity is not a neutral experience. It has consequences, sometimes for how people are treated, sometimes for eligibility for certain programs or benefits, sometimes for the community relationships that depend on shared identity. The Rh negative distribution is one of the traces of this complexity. When someone finds out they carry Rh negative blood and starts investigating why, they often end up in the middle of exactly these questions. Their Rh negative blood may reflect colonial European ancestry. It may reflect centuries of forced genetic mixing in the plantation South. It may reflect the founder effect in an isolated community whose full genetic complexity has never been publicly acknowledged. It may reflect pre-Columbian ancestry that the standard model of American history does not account for. It may reflect all of these things simultaneously in proportions that vary from individual to individual and from community to community. And following the trail of that investigation often leads into the parts of American history that official narratives most prefer to leave in the dark. Which brings us back to the title of this story. The darker reason is not singular. It is the accumulated darkness of all the things that American history did not want to face about itself. The violence, the mixing, the categorization systems designed to control populations rather than describe them accurately, the suppression of pre-Columbian complexity, the isolation that concentrated traits and tragedies simultaneously, the silences in the family records that DNA is now breaking open. All of this is why America has more Rh negative blood than almost any other country. Not because Americans are special, not because the blood itself is special, but because what happened here, the specific, violent, complicated, largely unexamined process by which this country came to contain the people it contains, left genetic signatures that other countries with different histories of population movement and mixing do not carry in the same pattern. The blood is a record, and reading that record honestly, with all the discomfort that honesty requires, is the real work of understanding why America looks the way it does on the global map of Rh negative blood. Let us follow one more thread before we begin to find our way toward the end of this story. It leads us into the southwestern United States and into a set of mysteries that have not received nearly enough attention in the context of this particular question. The desert southwest of North America, the region that now encompasses Arizona, New Mexico, Utah, Colorado, and parts of Nevada and California, was the site of one of the most sophisticated pre-Columbian civilizations in North America. The ancestral Puebloans, whom older literature sometimes calls the Anasazi, built cliff dwellings and great houses and astronomical observatories and road systems in the desert that survive as ruins today. Chaco Canyon in New Mexico was the center of a network of communities connected by roads that stretched hundreds of miles in straight lines across terrain that does not naturally favor straight lines. The Great Kiva at Pueblo Bonito in Chaco Canyon could hold hundreds of people. The astronomical alignments of Chacoan architecture are as precise as anything in the ancient world. And then, around 1150 CE, after several centuries of remarkable development, the Chacoan system collapsed. The great houses were abandoned. The roads fell into disuse. The population dispersed. Nobody knows exactly why. The drought record from Tree Ring shows that the region experienced severe and sustained drought in the century before the abandonment. But the Chacoan system had survived previous droughts. Something else may have contributed. Internal social collapse, external pressure, spiritual crisis, the kind of complex multi-factor breakdown that brings down systems that have grown too large and too complex for their resource base. The descendants of the people who built Chaco Canyon are the modern Pueblo peoples, the Hopi, the Zuni, the Rio Grande Pueblo. They carry oral traditions that speak of migrations, of coming and going over generations, of leaving places and returning to them, of the search for the center place. These traditions encode a real history of movement and resettlement that archaeology confirms. What the genetics of these communities shows is complex. There are markers of deep local ancestry, lineages that have been in the Southwest for thousands of years. There are also markers of longer-distance connections, genetic signatures that point to contact with populations from further afield. And in some Pueblo communities, there are genetic markers that have attracted attention because they do not fit straightforwardly into the expected ancestral picture. Some of these anomalous markers could reflect contact with Spanish colonizers who arrived in the Southwest beginning in the late 16th century. Some could reflect earlier contacts with other North American populations through the trade networks of the pre-Columbian period. Some cannot be easily explained by any of the currently documented contact histories. The Rh-negative frequencies in southwestern indigenous communities are generally low, consistent with the overall pattern of low Rh-negative prevalence in populations of Asian descent. But they are not quite as low as pure genetic drift from an exclusively Northeast Asian founding population would predict in some communities. And the anomalies cluster in ways that suggest some kind of additional genetic contribution, something that the standard model does not fully account for. This is a tentative observation. The data is not robust enough to support firm conclusions, but it adds to the overall picture of an American continent whose genetic history is more complicated than the prevailing models acknowledge, and whose Rh-negative distribution reflects that complexity in ways that are just beginning to be explored with the tools that modern genetics provides. There is one more historical figure worth meeting before this story is done. One more specific, human-grounded example of the way that the abstract patterns we have been tracing played out in the lives of actual people. Her name is not remembered because the records did not think her name worth preserving. She was an enslaved woman in South Carolina in the early 19th century, one of the millions of people whose individual lives are largely invisible to history, even though the collective impact of those lives shaped the genetic landscape of an entire country. What we know about her comes from the medical record kept by her enslaver, who was also a physician and who documented cases of medical interest on his plantation with the clinical detachment that characterized medicine of that era. In his records, he noted a woman who had lost 3 consecutive children in their first days of life. Each child was born alive and appeared healthy initially, then deteriorated rapidly, becoming jaundiced, lethargic, and anemic within the first 24 to 48 hours. None of them survived past the first week. He documented his attempts to understand the cause. His theories involved the mother's diet, the quality of the water, possible infections, the constitution of the children. None of his theories were correct. He was observing hemolytic disease of the newborn more than a century before the mechanism would be understood. What this record tells us is that the tragedy of Rh incompatibility was playing out in enslaved populations in America decades before anyone had any theoretical framework for understanding it. It tells us that the specific genetic circumstance created by the forced mixing of Rh-negative European genetic material with the previously Rh-positive West African gene pool of enslaved people was producing observable medical consequences within the first or second generation. And it tells us that those consequences were being documented not by people who were trying to understand them in the service of the people suffering from them, but by people who were primarily interested in the economic implications of infant mortality among enslaved populations. The physician's records continued with observations that are even harder to read. He noted that the woman was reproductively valuable, meaning that she was likely to produce children who would themselves be valuable as property, and that the loss of multiple infants was therefore an economic loss to be understood, and if possible, prevented. His concern was not for the woman or for the children who had died, but for the economic consequences of their deaths. And his inability to prevent those consequences, because he lacked the scientific understanding that would not be developed for another century, is documented in the same clinical detachment. This is one story, one physician's record of one woman's repeated losses. Behind it are millions more undocumented, unremarkable in the records of the people who controlled the documentation. Women who lost children without knowing why. Women whose bodies were at once being exploited for reproduction and sabotaged by the biochemical consequences of that exploitation. Women whose Rh-negative enslavers had introduced a genetic variant into the gene pool through violence, and whose bodies were now producing the medical consequences of that introduction in the deaths of the children produced by that violence. This is the human core of the darker reason. Not the genetics, not the statistics, the people, the specific, irrecoverable human lives that the history touched and often destroyed, and whose blood and whose bodies and whose losses are part of what produced the genetic landscape that researchers now observe and try to explain. I want to name that directly, because it is easy to discuss this material in the abstract, in the language of founder effects and population stratification and genetic drift, and to lose sight of the fact that behind every genetic data point is a human life. Behind every elevated frequency of a rare genetic trait in a particular community is a history of specific human beings making or being subjected to decisions that had consequences for their bodies and their children and their descendants. The woman in the South Carolina record was a real person. Her name was not preserved. Her story was documented only because it intersected with the economic interests of the person who owned her. But she was real. Her children were real. Their deaths were real. And the genetic consequence of the circumstances that produced them, the introduction of Rh-negative blood into the African American gene pool through centuries of exactly this kind of coercion and violence, is real in the bodies of living people today. That reality deserves to be held alongside all the genetics and all the history and all the archaeology. It deserves to be acknowledged as the human dimension of what would otherwise be an abstract story about blood types and migration patterns and founder effects. The science is real, and the history is real, and the human lives are real. And the most honest telling of this story holds all three together. Now, let us go somewhere unexpected. Let us go to the ocean. The North Atlantic in the last glacial maximum, approximately 20,000 years ago, was a different ocean than the one that exists today. The sea level was lower by more than 400 feet, exposing continental shelves that are now submerged. The coastline of Western Europe extended further into what is now the Atlantic Basin. The coast of North America extended further east, and the North Atlantic itself was partly covered by sea ice that extended further south than it does today, creating a continuous surface of frozen water along which, in theory, people with ice-adapted survival skills might have been able to travel. The people who might have traveled that route were the Solutreans of southwestern France and the Iberian Peninsula. We have discussed them briefly in the context of the Solutrean hypothesis of American pre-Columbian contact. Let us spend more time with who they actually were. These people, whose possible journey across the North Atlantic ice edge is one of the most debated questions in American archaeology, were a culture that flourished in southwestern Europe between approximately 22,000 and 17,000 years ago, during the depths of the last glacial maximum. They lived in some of the harshest conditions that modern humans have ever experienced outside of Antarctica, in a world of ice and cold and seasonal darkness that would challenge most modern people to endure for a single winter, let alone multiple generations. They were extraordinary craftspeople. The stone tools they produced, particularly the laurel leaf points that are their archaeological calling card, are among the finest examples of flint knapping in the entire prehistoric record. The best Solutrean points are so thin and so precisely shaped that they are nearly translucent. Flakes of flint work to such even thinness that light passes through them. This level of craftsmanship requires a combination of technical knowledge, physical skill, and what can only be described as artistic sensibility, a desire to make something more beautiful than mere function requires. They also left cave art, the paintings at Cosquer Cave in southern France, accessible now only by diving through an underwater entrance because the sea level has risen since the paintings were made, were created by people of the Solutrean period. The paintings show horses and bison and hand stencils, the same kinds of images found at other Ice Age sites across Europe. But the specific location of Kosker, on the Mediterranean coast rather than in the inland regions where most Solutrean material is found, is significant. It suggests that the Solutreans had a coastal dimension to their range and their economy that may have been more developed than the inland archaeological record shows. Coastal adaptation is crucial to the Solutrean hypothesis. The proposed migration route along the North Atlantic ice edge would have required precisely the skills that a coastal adapted population would have. Fishing, sealing, navigation along ice margins, the ability to survive in marine environments, and to travel along coastlines rather than overland. These are skills that leave few direct archaeological traces on land, because the camps and the equipment of coastal peoples tend to be located in places that are now underwater, swallowed by the sea level rise that accompanied the melting of the Ice Age glaciers. This is one of the reasons that the Solutrean hypothesis is so difficult to evaluate archaeologically. The most direct evidence for or against a North Atlantic coastal migration would be on continental shelves that are now 60 to 300 feet underwater. We cannot excavate them with current technology in any comprehensive way. We can find objects occasionally, fishing nets that bring up bones or artifacts from the seafloor, ROVs that can photograph and sometimes retrieve small samples from specific locations. But the systematic archaeological survey of the drowned coastlines where the evidence for or against pre-Columbian contact most directly exists is not something we can currently do. And so the question remains open, not answered, not definitively refuted, just open in the frustrating and fascinating way that questions become when the evidence that would resolve them is somewhere we cannot reach. The Solutreans, if they were the source of a pre-Columbian Atlantic migration to North America, came from one of the zones of highest Rh negative frequency in the world. The Iberian Peninsula and southwestern France, the territory where Solutrean culture was centered, is the same territory where modern Basque and related Celtic populations maintain the highest Rh negative frequencies in Europe. Whether this reflects continuity between Solutrean populations and modern Atlantic fringe populations, or whether the two distributions are related only indirectly through the general ancient genetic heritage of the region, we cannot say with certainty. But the geographic coincidence is striking. The people most likely to have attempted the kind of North Atlantic Ice Age crossing that the Solutrean hypothesis proposes were people from exactly the region where Rh negative blood is most concentrated in the world. If they made the crossing, they would have brought Rh negative blood to North America perhaps 20,000 years ago. And whatever genetic contribution they made to pre-Columbian American populations, however small and however diluted by subsequent Bering Land Bridge migrations, might persist as a faint signal in certain Eastern North American genetic profiles. That faint signal, if it is there, is one of the threads connecting the deep time story of Rh negative blood in America to the more recent historical layers we have been exploring. It is the possible ancient root of a pattern that historical circumstance then amplified in specific communities through specific mechanisms over the 5 centuries of documented American history. We cannot pull on this thread far enough to see where it ends. The evidence does not allow it, but we can see where the thread begins in the icy coastal margins of Ice Age Iberia. And we can trace it forward to the point where it disappears into the underwater archaeology and the uncertain genetics and the ongoing debates about who was in America before Columbus. And in that tracing, we add one more layer to the story of why America carries more Rh negative blood than almost any other country. A layer that predates Columbus by perhaps 20,000 years. A layer that may have been here before any of the other layers. A layer that connects the most ancient possible chapter of American prehistory to the same genetic trait that runs through all the more recent chapters. The blood has been here for a long time, possibly much longer than the official history acknowledges. and it has accumulated more depth and more complexity and more darkness with each chapter that has been added to the American story. That is where we are at the bottom of the deepest layer that this story reaches, looking up through 20,000 years of accumulated history at the surface of a present that is still in the process of discovering what happened on the way here. Something else needs to be said about what happens when genetic information and historical complexity intersect in the specific context of American race and identity. It is something that researchers in this field navigate with difficulty, and something that deserves direct acknowledgement in a story like this one. The information that modern genetics provides about American ancestry crosses lines that American society has historically maintained with great force. It reveals connections between communities that the law and the social order worked for centuries to keep separate. It demonstrates that the categories that American history used to classify people, categories that determined everything from legal status to access to education to the right to vote and to own property, were always fictions. Biological fictions useful to the people who controlled the categorization systems, destructive to the people who were categorized. When genetic data reveals African ancestry in families that identified as white, it is not revealing something that genetics invented. It is revealing something that those families and their ancestors worked very hard to conceal, because the consequences of disclosure in American society were severe and sometimes lethal. The fiction of white racial purity was maintained through social pressure, through legal enforcement, through violence, through the systematic destruction of evidence that contradicted it. The genetics survives because genetics does not respond to social pressure or legal enforcement or violence. It just persists generation after generation, quietly recording what actually happened. Similarly, when genetic data reveals European ancestry in African American families, it is not revealing something neutral. It is revealing the specific mechanism by which that genetic material was introduced, which was systematic sexual violence against enslaved women over a period of more than 2 centuries. The European genetic material in African American genomes is not the product of voluntary mixing between peoples who met as equals and chose each other. It is the product of coercion and rape and the systematic exploitation of enslaved women's reproductive capacity by their enslavers. This is one of the reasons why the American Rh-negative story is darker in a way that the simple global Rh-negative story is not. The elevated Rh-negative frequencies in African American communities carry within them the specific mechanism of their origin. They are a trace of violation, a biological record of what was done to enslaved women and their children, a record that persists in living people who may not know it is there or may not know what it means. This does not make the blood itself traumatic. People who carry Rh-negative blood do not carry any kind of curse or contamination. Blood is blood. Genetics is genetics. The people who carry this genetic heritage are simply people with all the dignity and complexity and worth that all people carry. But the story of how that heritage was created is a dark story. And understanding the full picture of American Rh-negative distribution requires sitting with that darkness rather than looking away from it. It also requires acknowledging something about how genetics has been used in American history, which is not always with the intention of revealing truth. Genetic and pseudoscientific arguments about racial difference have a long and ugly history in America. From the craniometry of the 19th century to the eugenics movement of the early 20th century to the intelligence testing debates that continue to the present day, scientific language has been used to dress up racial hierarchy in the clothes of objectivity, to make claims about the inherent differences between populations that are designed to justify differential treatment. The Rh-negative story can be hijacked by that tradition. The online communities that celebrate Rh-negative blood as the mark of a superior or specially gifted minority are participating, whether consciously or not, in a tradition of genetic exceptionalism that has always been dangerous. They are taking a genuine biological variation and inflating it into a marker of distinction, a basis for claiming special status or special knowledge or special connection to ancient and powerful lineages. This is not what the actual science supports, and it is not what the actual history of Rh-negative blood in America supports. The actual history, as we have traced it, is a history of violence and isolation and tragedy as much as anything else. The communities where Rh-negative blood is most concentrated in America are communities that did not choose their genetic heritage or their historical circumstances. They were shaped by forces they did not control, by violence done to them, by systems of classification designed to serve interests other than their own. Celebrating Rh-negative blood as a mark of special status in the context of that history is in its way a kind of insult to the actual story. It takes the genetic legacy of trauma and oppression and isolation and tries to turn it into a badge of honor, stripping away the human reality of what produced it. The actual honor, if that is even the right word, belongs not to the blood, but to the people who carried it through the circumstances that history imposed on them. The honor belongs to the enslaved women who survived and maintained family continuity despite everything done to shatter it. It belongs to the mountain communities that kept their traditions alive through generations of poverty and isolation. It belongs to the mixed-race communities that maintained community cohesion in the face of a society that refused to acknowledge their existence. It belongs to the people who crossed racial lines not out of cowardice but out of necessity, who built new identities to survive a system that would have destroyed them had they stayed where they were. Those stories are not about the blood. The blood is just the trace they left behind. The blood is just how we know they were there. Now let us step back from all the specific history and the specific genetics and spend some time with the larger question that this story illuminates. The question of what it means to inherit a complicated past in your body. Everyone carries history in their genetics. This is not unique to people who carry Rh-negative blood or to people with American ancestry or to people from any particular community. The genetic material that any individual carries is the cumulative product of every population event, every migration, every bottleneck, every instance of forced or voluntary mixing that affected their ancestors going back to the origin of the species. The specific traits that show up in any individual are a kind of geological cross-section through that history, a record of every layer through which their lineage passed. In most cases, the specific historical events that shaped a person's genetic inheritance are invisible to them. They do not know their ancestors survived a plague in 14th century Europe that killed most of the people around them. They do not know their ancestor was part of a founding population in a new territory whose small size amplified certain genetic traits. They do not know their ancestor crossed an ocean under conditions that would be incomprehensible to them today, carrying with them whatever genetic variants happen to be in their particular body. For Americans, and specifically for Americans from the communities we have been discussing, some of those invisible histories are darker than average. The founding events that shaped certain American gene pools were often violent. The migrations were often forced. The isolations were often imposed. The mixings were often coerced. And the genetic consequences of all of that are distributed throughout the living population as things that most people simply do not think about when they go to the doctor and get told their blood type. There is something strange and poignant about this. The body carries what happened. The blood remembers what the records forgot or were designed to obscure. And most of the time, the people whose bodies carry this history do not know it is there. They live their lives unaware that their cells are encoded with a record of events that happened centuries before they were born. Events whose consequences shape the world they live in, but that they have no direct access to or knowledge of. Genetic testing is changing this slowly, imperfectly, with its own set of complications and controversies. The genetics is making some of this hidden history visible. Families are discovering that the story they thought they knew is more complicated. Communities are finding that their genetic heritage contains contributions from populations that the official history said they never had contact with. Individuals are learning that the category they were assigned to by the social and legal systems of the country they live in does not accurately describe their actual biological ancestry. This is not comfortable. Truth often is not. But it is important because a country that does not know its own biological history, a country that has maintained fictions about its populations and their origins and their mixing and their separation, is a country that cannot fully understand itself. The genetics is one way of moving toward that understanding. Imperfect and partial, but real. There is something about the American Southwest that keeps drawing researchers back to questions that the standard model cannot answer. The landscape itself seems to demand unusual explanations. The enormous scale of what was built here in the pre-Columbian period and then abandoned. The complexity of the astronomical alignments. The mystery of the road systems that go nowhere that any contemporary map would find useful. The petroglyphs that cover cliff faces across thousands of square miles. Images that persist into the present without any consensus about their meaning. One of the most persistent mysteries is the presence of certain symbols in the rock art of the desert Southwest that appear to researchers who have studied ancient scripts from the Old World to bear resemblance to writing systems from across the Atlantic. Barry Fell, a Harvard marine biologist who became an amateur epigrapher in the latter part of his life, spent decades documenting what he claimed were Celtic Ogham inscriptions, ancient Phoenician texts, and other Old World scripts on rock faces across North America. His books were dismissed by mainstream archaeology, and some of his specific claims have been shown to be mistakes or misidentifications, but not all of them have been definitively refuted. And the question of whether some of what appears in American petroglyphs reflects genuine Old World contact has never been fully closed. What makes this relevant to our story is the geography. If there were pre-Columbian contacts between North America and the Atlantic fringe populations of Europe or North Africa, and if those contacts involve the same populations that carry the highest Rh negative frequencies in the world, then the presence of those populations' cultural signatures in American rock art would be additional evidence for the kind of contact that could have introduced Rh negative blood into North American populations before Columbus. This is speculative layered on speculative. The rock art is genuinely ambiguous. Mainstream archaeologists have legitimate reasons for skepticism about reading Old World scripts into images that may reflect purely indigenous American symbolic traditions. And even if some Old World script parallels in American rock art are real, they do not necessarily imply genetic contact at a scale that would leave a detectable signal in blood type distributions. But the questions are there. They persist the way all genuine mysteries persist. Because the evidence is suggestive without being conclusive. Because the anomalies pile up without quite reaching the threshold of proof. Because every time you think you have a clean explanation, something surfaces that complicates it. That persistence is itself informative. It tells us that the standard model is not fully adequate to explain what the evidence shows. It tells us that the history of North America before Columbus is more complicated than the textbooks present it, and that the complications have been systematically underexplored because of the political and institutional pressures that shape the development of American archaeology as a discipline. And in the space of that underexploration, the Rh negative question sits, one of many anomalies waiting for better tools and more honest inquiry. Let us make our way now to a kind of conclusion that is really an opening. Because the most honest thing this story can do is end not by closing down the questions, but by clarifying what they are, who they belong to, and what it would take to answer them. The question of why America has more Rh negative blood than almost any other country belongs in the most fundamental sense to the communities whose blood it is. To the African American families whose Rh negative frequencies reflect the specific history of what was done to their ancestors. To the Appalachian communities where isolation and founder effect concentrated rare traits alongside the particular cultures that isolation also produced. To the mixed race communities of the American South and East whose full genetic history has been systematically obscured. To the indigenous communities whose pre-Columbian genetics are still largely unknown, and whose post-contact genetic history reflects the specific catastrophe of epidemic disease and forced relocation and cultural disruption. These communities have not always had access to the scientific tools that would let them investigate their own genetic histories. They have not always had control over what was studied about them, who studied it, how the findings were reported, and what was done with the results. The history of scientific research on marginalized American populations is not a clean one. It includes exploitation, deception, the use of biological samples without informed consent, the publication of findings in ways that serve the interests of researchers rather than the communities they studied. That history is relevant to how any attempt to understand American Rh negative distribution needs to be conducted. Not as a reason to abandon the inquiry, but as a reason to conduct it differently, with the communities involved as partners rather than subjects, with the findings reported to the communities first, with control over biological samples remaining with the people who contributed them, with the questions being shaped in part by what the communities themselves want to understand about their own histories. This is the ethical dimension of the story, and it is inseparable from the scientific and historical dimensions, because the same communities whose blood carries the most historically complex and scientifically interesting patterns are also the communities that have the most reason to be cautious about outside researchers coming in to study them. Building the trust that would allow this research to be conducted ethically and collaboratively is slow work. It cannot be rushed. It requires sustained engagement with communities over time. The demonstration that researchers are genuinely committed to the community's interests and not just to the advancement of their own careers or the satisfaction of their own curiosity. It requires acknowledging the history of what previous research looked like, and being clear about how this research intends to be different. But that slow work is worth doing, because the questions are real, and the communities that hold the answers to those questions deserve to understand their own histories as fully as the science can illuminate them. And the country as a whole deserves to understand what its biological history actually looks like, rather than the simplified version that official narratives have substituted for the real thing. That is the final thing this story can offer. Not the answer to why America's Rh negative map looks the way it does, because that answer is too complex to be fully given, but the shape of the question and the recognition of who it belongs to, and the hope that the future will bring both better tools and better relationships that allow the question to be answered more fully and more honestly than it has been so far. The blood carries the history, and the history belongs to the people whose blood it is. And they deserve to know it in all its darkness, in all its complexity, in all the ways it connects them to ancestors whose stories were suppressed, and to events that shaped the world they live in, and to a lineage that stretches back further than any recorded history can follow. That is what this story has been trying to offer. That is the hope it ends on, in the quiet before sleep, in the particular darkness of a night that has been long and full and genuinely illuminating, in the way that honest engagement with difficult material always is. The blood moves, the record persists, and somewhere in the distance, the questions keep waiting for the answers they deserve. There is one more place this story needs to go before it can find its way to something like a conclusion, and it is back to the very beginning of the American project, to the period before the first colonial settlements, to the centuries immediately before European contact, when the continent that would become the United States was inhabited by millions of people whose complex histories and diverse cultures were about to be transformed by forces they had no way of anticipating. The pre-Columbian population of North America has been estimated at between 8 million and 18 million people, depending on the methodology used and the region considered. These were not uniform populations. They were as diverse in language, in culture, in technology, in social organization, and in genetic ancestry as any comparable geographic region anywhere in the world. The languages alone numbered in the hundreds. The cultures ranged from the agricultural city builders of the Mississippi Valley to the maritime peoples of the Pacific Northwest coast, to the nomadic hunters of the Great Plains, to the desert farmers of the Southwest, to the woodland peoples of the East, each with their own sophisticated adaptation to their particular environment. These populations had been in contact with each other for thousands of years through trade networks that span the continent. Goods moved from the Gulf Coast to the Great Lakes. Copper from Lake Superior showed up in burial mounds in Florida. Obsidian from Yellowstone appeared in archaeological sites in Ohio. Shells from the Pacific coast moved into the interior. Ideas moved along with the goods, and so did people, and so did genetic material. The genetic diversity of pre-Columbian North American populations was accordingly complex. Different communities had different founding histories, different levels of isolation and contact with neighboring groups, different proportions of the various genetic variants that had accumulated in their ancestral populations over the millennia since their ancestors first arrived in the Americas. The picture was never the simple, homogeneous one that later centuries of European imagination would construct. When epidemic disease arrived in the wake of European contact, beginning in some regions before any European explorer personally visited them, spread along trade routes by the first infected contacts, the impact on this complex, diverse, multiply-connected population was catastrophic in ways that are difficult to fully comprehend. Entire communities were eliminated. Languages died without being recorded. Cultural practices that had been maintained for generations were disrupted beyond recovery. The trade networks that had connected communities across the continent and maintained gene flow between them were broken. And the survivors, the people who happened to have whatever genetic resistance to the specific pathogens that hit their community, were in many cases reduced to such small numbers that the subsequent recovery of those communities drew from radically restricted genetic pools. The founder effect operated again in the aftermath of the epidemics, concentrating whatever genetic variants had survived in the small populations that managed to reconstitute themselves. If any of those pre-Columbian communities carried Rh-negative blood, whether from ancient transatlantic contacts or from other mechanisms we have discussed or from causes that we have not yet identified, the epidemics would have been one more filter on that genetic heritage. Communities were reduced to remnants. Those remnants intermarried among themselves, and sometimes with European and African-descended populations as the colonial period advanced. And the genetic material that survived those multiple filters, the epidemics, the forced relocations, the assimilation pressures, the intermarriage with colonial populations, is what we see today in the DNA of Native American communities and in the DNA of the many mixed ancestry communities whose Native American heritage is often unacknowledged or unknown. In this sense, the Rh-negative frequencies we observe in certain communities with indigenous American heritage are not just a reflection of post-Columbian European contact. They are also a reflection of the specific dynamics of survival and bottleneck and reconstitution that the indigenous populations of North America went through in the centuries after European contact. They reflect which communities survived and in what numbers and who they subsequently mixed with as the colonial and post-colonial periods proceeded. The genetic record of indigenous American survival is one of the least studied and least understood aspects of American genetics. There are political reasons for this, having to do with the fraught relationship between genetic research and indigenous communities who have had experiences with scientific institutions that give them legitimate reasons to be cautious about contributing biological samples. There are logistical reasons having to do with the complexity and diversity of indigenous American genetic backgrounds. And there are historical reasons having to do with the simple fact that the documentation of indigenous genetic history was not a priority for the institutions that controlled research funding until very recently. The result is that we know less about the genetic history of indigenous American populations than we know about almost any other group of comparable size and historical significance. And this gap in our knowledge is reflected in the incompleteness of the American Rh-negative story. We do not fully know what pre-Columbian frequencies were, We do not fully know what survived the epidemics. We do not fully know what the admixture patterns were in the colonial and post-colonial periods for communities that were simultaneously losing members to disease and displacement and gaining genetic material from European and African-descended populations through various mechanisms. What we do know is that the picture is complicated, that the genetics of indigenous American communities in the present does not straightforwardly represent the genetics of pre-Columbian North America because too much happened in between, and that whatever Rh negative frequencies we observe in those communities today reflect a history that includes not just the deep time origin stories, but also centuries of epidemic death and forced movement and cultural disruption and genetic mixing that reshaped everything. This is the part of the American Rh negative story that is hardest to tell, because the evidence is thinnest and the history is most painful, and the people whose history it is have had the least say in how it is told. The story of what happened to the indigenous peoples of North America in the aftermath of European contact is a story of loss so total and so specific that it barely registers in the American national consciousness as something that happened to real people with real bodies and real genetics and real descendants who are still here, carrying that history in them. Whether they know it or not, they are here. The descendants of the people who lived through the epidemics and the removals and the boarding schools and the assimilation campaigns are here. Some of them know their indigenous ancestry. Some of them, through the various mechanisms by which indigenous identity was suppressed and hidden over the centuries, do not. Some of them are included in the federal recognition system for Native American tribes. Many are not. Some carry genetic markers that point unambiguously to indigenous American ancestry. Others carry markers that reflect the complex mixing of the post-contact period and are therefore less straightforwardly interpretable. All of them carry the history in their blood, and for some of them that history includes whatever Rh negative blood was present in the pre-Columbian populations of North America, carried forward through all the filters of survival and bottleneck and mixing that the subsequent centuries imposed, arriving in the present as one more trace of a past that the official story of America has never fully acknowledged. And here's the thing about all of this. Here is what you take with you into the quiet when this story is over. The blood does not know about official histories. It does not obey census categories or racial classification systems or the legal definitions of who belongs to which group. It just moves through bodies and passes from parent to child and carries what it carries, regardless of what anyone says about it or thinks about it or tries to prevent it from being. The Rh negative blood in America carries all of this history. The European Ice Age heritage, the colonial settlement, the forced mixing of the plantation system, the founder effect in isolated communities, the racial passing, the indigenous survival and reconstitution, the pre-Columbian mysteries, the deep time bottleneck that shaped the human species nearly 1 million years ago. It carries all of it simultaneously in all the people who carry it, in proportions that vary from individual to individual and community to community in ways that reflect the specific historical circumstances that shaped each particular lineage. That is extraordinary, not in a mystical sense, not in the sense that the blood confers special powers or marks its carriers as chosen or connects them to ancient divine lineages, but extraordinary in the straightforward sense that the human body, in this as in so many other respects, turns out to be a more complete and more honest archive of what actually happened than any document or record or official narrative that human beings have constructed. The blood tells the truth about migration, about trauma, about survival, about the mixing and the violence and the isolation and the choices made and the choices that were never choices at all. About the long, dark, complicated, astonishing history of how the people who live in America came to be where they are and carry what they carry. That is why this question matters. Not because Rh negative blood is special in itself, but because asking why it is distributed the way it is in America leads you, if you follow the question honestly, into the most important and most suppressed and most real parts of the American story. And that story deserves to be told in full, in the dark, slowly, and with patience. For everyone who is lying here listening and wondering what they are made of and where they came from, and what it means that the world turned out to be so much more complicated and so much more connected than anyone told them it would be. Before we move toward the final stretches of this story, let us pause and consider what we have not yet addressed. Because there are dimensions of the American Rh-negative question that exist in stranger territory than any of the documented history we have covered, and they deserve to be approached with the same mix of intellectual openness and critical honesty that the rest of the story has required. There are oral traditions among certain Native American groups that speak of ancient visitors, of people who came from across the Great Water before the coming of the Europeans. People who were different in appearance and in knowledge, who stayed for a time and left their marks, some of which are still visible in the landscape, and some of which are no longer visible except in the genetics of the descendants of those who encountered them. These traditions are not evidence in the scientific sense. They are not documents that can be cross-referenced with other records. They are stories, and stories are shaped by the purposes they serve and the communities that maintain them and the generations of telling and retelling that transmit them across time. They cannot be taken as literal historical accounts in the way that archaeological data can, but they are not nothing. The oral traditions of indigenous peoples have been consistently undervalued by Western scholarship, which has tended to treat written records as the only legitimate form of historical documentation. And that undervaluation has repeatedly turned out to be wrong. Oral traditions that scholars dismissed as myth have turned out to encode real historical events. The Aboriginal Australian traditions of rising sea levels, dismissed for decades as symbolic or mythological, have been validated by geological evidence of exactly those sea level changes occurring at exactly the times and in exactly the places the traditions describe. The traditions are real records of real events transmitted with remarkable fidelity across thousands of years through mechanisms that Western scholarship is only beginning to understand. If some North American oral traditions contain accounts of pre-Columbian contact with people from across the Atlantic, accounts that have been maintained through the same kind of intergenerational transmission that preserved accurate information about sea level changes, then those accounts deserve serious attention, even in the absence of corroborating physical evidence. They may be describing something that happened, and if they're describing something that would have had genetic consequences, including potentially the introduction of Rh-negative blood into certain North American populations at a time, and through mechanisms that the standard history does not account for. This is where the story becomes genuinely uncertain and genuinely interesting. The documented history is dark, but comprehensible. It involves forces and mechanisms that are well understood, even if their full implications have not been publicly acknowledged. But the pre-documented history, the period before writing, before records, before the instruments that modern scholarship depends on, is a different kind of darkness. It is the darkness of genuine unknowing, of things that may have happened but cannot be proven, of patterns in the evidence that suggest something without proving what that something was. American archaeology has a troubled relationship with this kind of uncertainty. The field has at various points in its history been eager to find connections between pre-Columbian American civilizations and Old World cultures. Sometimes because of genuine interest in the question, and sometimes because of less admirable motivations, including the desire to attribute the achievements of indigenous American peoples to outside influence rather than indigenous genius. The history of that eagerness and the bad science and worse politics that sometimes accompanied it has made mainstream American archaeology cautious to the point of excessive skepticism about any suggestion of pre-Columbian contact. That caution is understandable, but it has costs. It creates a field where anomalous evidence tends to be suppressed or dismissed rather than carefully investigated. It creates a situation where the burden of proof for claims of pre-Columbian contact is set so high that even genuinely suggestive evidence cannot get a fair hearing. And it creates a public vacuum that is filled by the less scrupulous end of the alternative history community, which seizes on every anomaly and spins it into confirmation of whatever grand narrative happens to be fashionable at the moment. The result is that the most interesting and most contested questions about American prehistory tend to be discussed either in extremely cautious and qualified academic language that most people will never read, or in breathless alternative history language that strips away all the qualifications and presents speculation as fact. Neither extreme serves the goal of actually understanding what happened. What this story has tried to do is occupy the middle ground, to take the anomalies seriously without inflating them, to acknowledge the uncertainty without using it as license for wishful thinking, to follow the evidence as far as it goes, and then clearly mark the point where evidence ends and hypothesis begins. That middle ground is uncomfortable. It does not offer the clean satisfaction of a proven explanation or the dramatic excitement of a sensational revelation. But it is where the most interesting and most honest inquiry lives. And in that middle ground, the American RH negative question sits with its many layers and its multiple explanations and its genuine mysteries. The documented layers are darker in documented ways. The undocumented layers are uncertain in ways that may never be fully resolved. And all of them together make up a picture of a country that does not fully know its own biological history, that carries in its population genetic traces of events it has not fully reckoned with, and that is only beginning to develop the tools and the will to look honestly at what those traces actually mean. Now let us travel to the final and perhaps most profound dimension of this story. The one that connects all the historical and genetic particularity of the American case to something universal about the human condition and about what it means to carry the past in your body. Every human being alive today is the product of an unbroken chain of survivors. Every single one of your ancestors, going back to the first members of our species 300,000 years ago, and back through them to our earlier ancestors, and through them all the way to the first cells that figured out how to replicate themselves more than 3 billion years ago, every single one survived long enough to reproduce. Not one of them died young enough to fail to pass their genetic material forward. The chain is unbroken. In the face of ice ages and droughts and epidemics and predators and violence and all the other things that have killed individuals and sometimes populations across the long history of life on Earth, your particular lineage survived. All of it. Every link in the chain. That is astonishing when you actually sit with it. Not special to you or to RH negative people or to any particular population. Universal. Every person alive carries that same unbroken chain of survival. But the specific events that chain passed through are different for different lineages. And those specific events left marks in the genetics that persist in the present. For the lineages that contributed to the American RH negative pool, the chain passed through some particularly dark and specific events. The bottleneck that compressed the entire human lineage to 1,000 individuals nearly 1 million years ago. The ice ages that concentrated certain populations in Atlantic fringe refugia and selected for specific genetic profiles in those populations. The ancient migrations, whether documented or not, that carried people and their genetics across oceans to new continents. The epidemics that devastated pre-Columbian North American populations and compressed surviving gene pools to remnants. The transatlantic slave trade that moved millions of people and their genetics across an ocean against their will. The plantation system that forcibly mixed genetic material through systematic violence. The isolation that concentrated rare traits in small communities, and the marginalization that maintained that isolation across generations. The racial passing that distributed genetic material across official boundaries. All of these events across all of this time contributed to the specific genetic heritage that American RH negative people carry. That heritage is complicated. It contains both beauty and horror. It contains the genetic legacy of ancient peoples who survived ice ages that should have killed them, who crossed oceans that most of their contemporaries never approached, who built civilizations in environments that would challenge most modern people to simply inhabit. And it contains the genetic legacy of violence and coercion and exploitation, of the specific darker chapters of American and human history that produce specific biological consequences in specific communities. To carry this heritage is not to carry a burden or a distinction in any simple sense. It is simply to carry what you carry. To be what your ancestors made you through their surviving and their mixing and their passing forward of whatever they had to pass forward. The complexity and the darkness of the story that produced that heritage does not diminish the people who carry it. It does not define them or limit them or explain them in any complete sense. But it is worth knowing. It is worth following the thread back as far as it will go, through the documented history and into the undocumented, through the genetics that can be tested and into the mysteries that cannot yet be resolved. Because the knowing, even when it is incomplete, even when it leads into darkness rather than light, connects living people to the full reality of their origins in a way that the simplified stories cannot. And the simplified stories have costs. When a country tells itself a simplified story about who its people are and where they came from, it loses the ability to understand certain things about itself. It cannot account for the anomalies in the data. It cannot explain the genetic traces of histories it refuses to acknowledge. It cannot fully understand the communities that formed in ways that the simplified story does not include. And it cannot honor the actual ancestors whose actual lives and actual suffering and actual survival produce the actual people who are alive in that country today. The American RH negative story, told fully and honestly, is a way of recovering some of what the simplified story lost. It is a way of saying, here is what the blood actually shows. Here is what the genetics actually indicates. Here is what happened, as best we can tell. And here is what remains unknown and uncertain. And here is why it matters that we try to know, even when the knowing is uncomfortable, even when it opens doors onto history that the official narrative preferred to keep closed. Let us go to one more specific story, one that crystallizes all of these abstractions into something concrete and human and still largely unknown to the broader American public. In the autumn of 1996, a pair of college students in Kennewick, Washington, discovered a skull in the shallows of the Columbia River. They called the police, who called a forensic anthropologist, who determined that the skull was old, very old. The remains were subsequently dated to approximately 9,300 years before the present, making them among the oldest, most complete human skeletons ever found in North America. The skeleton became known as Kennewick Man, and it immediately became the center of one of the most contentious and revealing controversies in the history of American archaeology. The problem was the skull. It did not look like what researchers expected from a 9,300-year-old North American skeleton. The morphology, the shape of the cranium and face, did not match the morphological profiles of known ancestral Native American populations, which are associated with Northeast Asian origins. Instead, it showed features that some researchers described as resembling European or even Polynesian populations more closely than Asian ones. This finding triggered an immediate and intense dispute that played out simultaneously in the scientific literature, in the courts, and in the arena of American Indian political advocacy. Several Native American tribes, asserting that any ancient human remains found in their traditional territory should be considered their ancestors and should be subject to their control for purposes of reburial, sought to have Kennewick Man returned to them under the Native American Graves Protection and Repatriation Act. Scientists who wanted to study the remains argued that the remains were not demonstrably associated with any existing tribe, and that returning them for reburial before complete study would result in the permanent loss of irreplaceable scientific information. The legal battle lasted a decade, during which time the remains were held by the Army Corps of Engineers, while scientists conducted limited studies with court permission. The final ruling allowed scientific study to proceed, and subsequent genetic analysis confirmed what the morphological study had suggested. The genetic profile of Kennewick man was not straightforwardly consistent with ancestry in the populations that would be expected from a pure Bering Land Bridge migration model. The closest living population to Kennewick man genetically appeared to be the indigenous peoples of Polynesia, specifically certain populations from the Ainu of Japan and from the native peoples of the Pacific Islands. This finding does not prove transatlantic contact. It does not prove any specific alternative theory of American prehistory, but it does confirm that the human population of North America 9,000 years ago was more genetically diverse and more genetically complex than the standard model predicts. There were people here whose ancestry pointed in directions that the textbooks had not accounted for. Where those people came from, how they got here, and what happened to them and their genetic contribution in the millennia that followed, these questions have not been fully answered. They may not be answerable with current evidence. But the fact that a 9,300-year-old man found in the shallows of a river in Washington state had a genetic profile that did not fit the expected pattern is itself important information. It tells us that the story of North American prehistory is more complicated and more open than most people have been told. And it connects to the Rh-negative question in a way that is hypothetical but worth stating. If the ancestral American population 9,000 years ago included people whose genetic profiles pointed toward Polynesia, toward the Pacific Islands, toward ancient populations that are themselves ancestrally connected to the complex mixing patterns of early modern humans in Southeast Asia and the Pacific, then the genetic diversity of that ancient American population was greater than we usually assume. And genetic diversity means the presence of a wider range of genetic variants, including potentially whatever variants were present in the ancient populations that contributed to that diversity. We do not know whether the ancestral population that Kennewick man represented carried Rh-negative blood. The Rhd gene status of Kennewick man has not been reported in the scientific literature. But the existence of Kennewick man and of the genetic complexity he represents reminds us that we should not assume we know what the pre-Columbian genetic landscape of North America looked like. We have far fewer data points than we need, and the data points we do have keep suggesting that the picture was more complicated than the standard model. In that space of genuine uncertainty, the questions about American Rh-negative blood remain open. They remain interesting, and they remain connected to the larger questions about American identity and American history that this country has never fully resolved. Now let us spend some time thinking about what it means to live in the aftermath of all of this. To be alive in America in the present moment, carrying whatever genetic heritage you carry, in a country that is still in the process of deciding how to understand and reckon with the history that produced its current population. The conversations about race in America have been getting louder and more urgent over the past decade. The data from genetic testing services has been quietly complicating those conversations by making it harder to maintain the fiction of distinct racial categories with clearly bounded genetic identities. Millions of Americans have discovered that their genetic ancestry is more complicated than their social identity. And that discovery has rippled through families and communities and the wider culture in ways that are still playing out. The Rh-negative distribution is one small thread in that larger unraveling. It is one of the places where genetics tells a more complicated story than the official categories allow. It is one of the data points that suggests the real history of America's population, the biological history, is not the same as the story that official history constructed and maintained. This matters not because blood determines identity, and not because genetics is destiny, and not because the complexity of American genetic heritage implies any particular political conclusion. It matters because truth matters, because understanding what actually happened is important regardless of whether the understanding is comfortable, because the people whose stories are encoded in the American genome deserve to have those stories told, even the parts of them that the country has preferred to forget. The darker reason why America has more Rh-negative blood than almost any other country is at its core a story about what happens to genetics when history is dark. when populations are forced to move and mix and isolate in ways that they did not choose, when the official record is designed to obscure rather than document what actually happened, when survival requires passing through filters so severe that only certain lineages make it through. When the country that emerges from all of this continues to tell itself a simplified story that cannot account for the complexity that its own bodies carry. The blood carries the truth. It always has. And increasingly, we have the tools to read what it says if we are willing to look and willing to sit with what we find. We are at that point in the story where the threads begin to gather back together, where the journey that has taken us from the biochemistry of a single protein to the deepest mysteries of human prehistory begins to find its way back to the present. To the people alive right now who carry this history in them without necessarily knowing it is there. If you are lying in the dark listening to this, you are in that present. You are one of the people who carries whatever history your particular lineage passed through. Maybe you are Rh negative and have been listening for the specific resonance that this story might have with your own genetics. Maybe you are Rh positive and have been listening because the questions about where we come from and what the blood remembers are questions that belong to all of us, regardless of blood type. Maybe you are somewhere in the middle of figuring out what your ancestry is and what it means and how the official story you were told about yourself matches or doesn't match what the biology actually shows. Wherever you are and whatever you carry, this story is part of the larger context of what it means to be human in general and what it means to be American in particular. The blood that moves through you right now has been moving for 300,000 years or more, passing through every crisis and every miracle and every darker chapter that your particular ancestral lineage survived. It arrived in America, if it is American blood, through pathways that were often dark. And it has been shaped by American history into a profile that reflects all the complicated, suppressed, and genuinely unknown dimensions of that history. That is remarkable, not in a way that sets any particular person or community apart, but in the universal way that every human body is remarkable as an archive of survival that stretches back further than any recorded history and forward into futures that none of us can see. The question of why America carries more Rh negative blood than almost any other country is ultimately a question about who Americans are and how they came to be here and what it cost and what it produced. It does not have a single clean answer. It has many answers layered on top of each other, darker in various ways, extending across time scales from the human to the geological. But asking the question honestly, following it wherever it leads, and sitting with the discomfort of what it reveals is one of the most important things a country can do. Because a country that cannot understand what its own bodies carry cannot fully understand itself. And a country that cannot understand itself cannot do the work that its history requires. There is a particular quality to the darkness just before dawn. It is different from the darkness of midnight. Midnight darkness is full and complete, settled and total. But the darkness just before dawn has something in it. A faint awareness of what is coming, a sense that the night has been moving towards something, that all the hours of darkness have been carrying the day forward. This story has been that kind of darkness, not the darkness of despair or of things hidden for the purpose of hiding them, but the darkness of things that need to be seen clearly before they can be properly understood. Things that require the patience to sit with what is difficult and what is unknown and what has not yet been resolved. We have covered a great deal of ground, from the discovery of the Rh factor in a New York laboratory in the 1940s to the Ice Age refugia of the North Atlantic Fringe. From the plantation systems of the antebellum South to the isolated hollows of Appalachian mountain communities. From the pre-Columbian civilizations of the Mississippi Valley to the contested genetics of ancient remains found in Washington State. From the biological mechanism by which Rh incompatibility kills babies to the epigenetic mechanisms by which intergenerational trauma alters physiological baselines in descendants. From the documented history of racial passing and the deliberate concealment of ancestry to the undocumented possibility of pre-Columbian transatlantic contact that would push the introduction of Rh-negative blood into North America back thousands of years before Columbus. Through all of it, we have been following the same question. Why does the United States carry elevated Rh-negative frequencies that do not straightforwardly reflect the genetic history that official narratives describe? And the answer, as we have found, is not one thing. It is a layering of explanations, each darker in its own way, each reflecting a different dimension of the complicated and often violent history by which America came to contain the people it contains. The European colonial immigration that brought Atlantic fringe genetics to a new world. The forced movement of enslaved Africans and the systematic mixing of their genetics with European genetic material through coercion and violence. The isolation of mixed-race and marginalized communities that concentrated rare traits through the founder effect. The racial passing that distributed genetic material across official boundaries in ways that were invisible to official record. The suppression of pre-Columbian complexity and the questions about ancient contact that the mainstream model has not satisfactorily resolved. The deep time legacy of Ice Age survival and near-extinction bottlenecks that shaped the global distribution of Rh-negative blood before any of the specifically American history began. None of these explanations is comfortable. Together they paint a picture of a country whose genetic reality was always more complicated, more mixed, more dark in its origins than the simplified narratives of national identity prefer to acknowledge. A country where the blood tells a different story than the census, where the genetics record what the records were designed to conceal, where the body is a more honest archive than any official document. This is not a reason for despair. It is a reason for honesty, and honesty about the complicated, dark, and genuinely fascinating story of how American blood came to be the way it is one of the ways that a country can begin to understand itself more fully and therefore to do better by the people whose bodies carry all of that history. The blood is there in millions of American bodies, in people who know their blood type and have wondered about it, in people who have never thought about it, in people who know nothing about the history we have traced tonight, and in people for whom parts of this story are intimately familiar. It carries what it carries, regardless of whether anyone knows or acknowledges or understands what it means. But meaning can be found. History can be recovered. The genetics is one key, imperfect and partial but real, to parts of American history that were deliberately locked away. Following the blood honestly leads to places that official narratives do not go. And those places are important, not because the blood makes anyone special, but because the history it encodes belongs to the people who carry it and to the country that was built by all of those people. And that history deserves to be told. One more thing before we begin the very last stretch of this journey. It is a small thing, but it matters. The families in isolated Appalachian communities who lost babies to Rh incompatibility generation after generation without knowing why. The enslaved women whose bodies were violated and whose children carried European genetic material in addition to their own, children who were enslaved like their mothers. The mixed-race communities that maintained their internal cohesion despite a society that refused to acknowledge their existence. The people who crossed racial lines and built new lives and new identities, carrying their genetics with them into communities that did not know what they were receiving. The indigenous survivors of epidemics who reconstituted their communities from remnants and continued to maintain cultural continuity despite everything that had been done to break it. All of these people deserve to have their stories told, not to elicit sympathy, though the circumstances of their lives often merit it, not to assign guilt, though the systems that created those circumstances certainly deserve scrutiny, but simply because they were real, and what happened to them was real. And the consequences of what happened to them are still real in the genetics of the people alive today, and they deserve to be seen. The blood sees them even when the official history does not. And this story, for all its length and all its complexity and all its darkness, is ultimately in service of that seeing. Now we are in the very last part of the long journey tonight. And it seems right to slow down here, the way any long journey slows as it approaches its destination, the pace naturally settling into something more contemplative than the ground-covering momentum of the middle miles. You have been traveling with this story for a long time now, through documented history and speculative history and deep time and the particular painful specificities of American identity and American genetics. Through the biology of a single missing protein and its enormous consequences for pregnancy and for survival and for the reproduction of isolated communities. Through the mystery of ancient contacts and the question of what was here before the historical record begins, and the possibility that the blood carries traces of things that happened before anyone was writing anything down. And now the journey is settling, not resolving into a clean answer, because clean answers are not what the evidence provides, but settling into the particular kind of rest that comes after a long and honest engagement with difficult material. The kind of rest where you know more than you did, and the knowing is not entirely comfortable, but feels in some deep sense more real than the comfortable simplifications it replaced. The blood is there. It has been there through all of this, moving through bodies, carrying its history, indifferent to the stories we tell about it, but shaped by the events those stories describe. It will continue to be there, passing from parents to children, being tested and studied and gradually better understood, carrying the record of everything that happened forward into a future that will know more about it than we do. And the question we began with, the darker reason why America carries more Rh negative blood than almost any other country, remains open in some of its dimensions. The documented layers are darker but clear. The undocumented layers are uncertain in ways that may or may not yield to future investigation. The deep time layer is ancient beyond any complete understanding, but visible in the patterns of genetic variation that extend backward through the human story farther than history can follow. What the question has given us in the asking and the pursuing of it is something like a map, not a complete map, not a map that tells you with certainty where everything is and how it all connects, but a map that shows you the terrain, that marks where the documented roads run and where they end, that indicates the territory beyond the roads where the evidence suggests something might be but cannot yet be fully seen, and that is honest about which parts of the terrain remain genuinely unexplored. That is what following a mystery honestly gives you. Not resolution, but orientation. Not all the answers, but a better sense of where the answers might be found and what the questions actually are. Let us now turn to something that has been waiting in the background of this whole story. Something that puts all the history and genetics into a different kind of perspective. The perspective of what the future might reveal. We are living through the early decades of the genomic revolution. The tools that allow us to sequence human DNA, to trace genetic lineages with precision, to analyze ancient remains and extract information about populations that lived thousands of years ago. These tools did not exist in any serious sense 30 years ago. They have improved exponentially since then and continue to improve. The amount of human genetic data available for analysis has grown from thousands of samples to tens of millions, and it grows every year. This expanding data set is changing what we know about human prehistory at an accelerating rate. Each year brings new findings that complicate or overturn previous models. Ancient DNA extracted from remains that were previously considered too old or too degraded for meaningful analysis is revealing genetic patterns in ancient populations that nobody predicted. The analysis of existing modern genomes with new computational tools is finding signals that previous methods missed. The integration of genetic data with archaeological and climatic and linguistic data is producing increasingly detailed pictures of human movements and mixings and separations across the deep past. For the American Rh negative story specifically, this means that the uncertainties we have been sitting with may not be permanent. Some of the questions that cannot currently be answered about pre-Columbian contact histories, about the genetics of pre-epidemic populations, about the ancestry of the isolated communities that carry anomalous genetic profiles, may become answerable as the science develops. Ancient DNA from pre-Columbian American burial sites, properly consented and ethically collected in collaboration with indigenous communities, could provide direct evidence about what Rh negative frequencies looked like in North America before European contact. Better analysis of the genetic profiles of isolated mixed-race communities could reveal the specific contributions of different ancestral populations and potentially identify genetic traces of contact histories that are not currently documented. This is not guaranteed. Science does not always deliver the answers that the questions seem to demand. Some things remain uncertain even as the tools improve. But the trajectory is toward more knowledge rather than less. And that trajectory is one reason to hold the current uncertainties with patience rather than frustration. The other reason to hold them with patience is that some of what matters most about this story is not the specific answers to specific genetic questions. It is the practice of asking them honestly, the practice of looking at the American genetic landscape and taking seriously what it shows rather than looking away, because what it shows complicates the preferred narrative. The practice of following the evidence, even when it leads into the parts of American history that the official story preferred not to examine. That practice is valuable regardless of what the specific answers turn out to be. A country that is willing to look honestly at the genetic evidence of its own history is a country that is capable of reckoning with that history in other ways as well. A country that dismisses genetic anomalies because they complicate the official narrative is a country that will also dismiss historical evidence and cultural evidence and the testimony of the communities whose experiences are encoded in those anomalies. The blood is asking questions that American history has not fully answered. Listening to those questions, even when the listening is uncomfortable, is part of what it means to take the past seriously and to take the people who lived through it seriously. There is also something to be said about what it feels like to carry a mystery in your body. To know that your blood belongs to a small global minority of people who lack a protein that virtually every other primate on Earth has. To feel the weight of that difference, even though the difference is invisible and has no behavioral or cognitive consequences, even though the only times it directly matters are in medical contexts related to blood transfusions and pregnancy. The feeling is real, even when its substance is primarily symbolic, and the symbolism is connected to real things. The feeling that your blood marks you as somehow set apart connects, whether you know it consciously or not, to the actual history of the lineages that carry this trait, to the actual isolation of the Atlantic fringe populations that concentrated it, to the actual violence that introduced it into communities that did not originally carry it, to the actual mysteries about its origins and its possible pre-Columbian presence in North America. The symbol is pointing at something real, even if what it is pointing at is more complicated and darker and less flattering than the ancient alien theories and divine bloodline narratives would suggest. The real story is stranger than the mythological one. It is stranger because it involves actual events, actual people, actual consequences. It is stranger because it reaches into the deep past of human evolution and into the near past of American history and connects them through a single biochemical trait. It is stranger because it reveals a country that does not fully know what it is made of, and a species that does not fully know what happened to it on the way to the present. And in that strangeness, in the space between what we know and what we can only infer and what we cannot yet know at all, there is the particular quality of mystery that belongs specifically to the very old and the very human. The quality of sitting in the dark and knowing that the darkness holds things that the light has not yet reached. Things that were real and that mattered and that left their marks. Waiting for the tools and the patience and the honesty to finally find them. Something else needs to be acknowledged before we end. Something about how this story feels. Not what it means analytically, but what it feels like to travel through it over the long arc of a night like this one. There is grief in this story. The grief of communities that lost children without knowing why. The grief of peoples whose cultures were shattered and who maintained continuity against enormous odds. The grief of histories that were deliberately suppressed and of people whose real stories were replaced by the stories that a particular social order found more convenient. The grief of the unknown, of the things that happened and left no record except the blood. And even the blood does not fully say what it knows. There is also something that is not quite the opposite of grief, but that coexists with it. Call it wonder. The wonder of a species that survived a near-extinction event nearly 1 million years ago and rebuilt itself from a genetic thread so thin it seems impossible. The wonder of human beings crossing oceans before anyone thought it was possible, following the stars and the birds and the waves into unknown territory, and arriving and surviving. The wonder of isolated communities maintaining themselves against all the forces that would have liked to see them assimilated or destroyed, preserving whatever they had to preserve, passing it forward to children who would do the same. The wonder of the blood itself, this ancient biochemical system that has been moving through human bodies for 300,000 years, carrying the record of everything that happened along the way. Grief and wonder together. That is the emotional texture of this story. And it seems right that it should be both. Real history engaged with honestly always is both. Let us make our way toward the very end now, but slowly, without rushing, because the end of a story like this one should feel like the end of a long walk. The kind of walk where you arrive somewhere and look back and can see the whole of the distance you have covered. All the varied terrain, the darker passages and the open stretches, the places where the path was clear and the places where you were finding your way by something other than sight. The question we started with has not been answered cleanly because it does not have a clean answer, but it has been opened up in ways that should feel more satisfying than a clean answer, because the opening is closer to the truth than any simplification would be. America has more Rh-negative blood than almost any other country, for reasons that include colonial European immigration from Atlantic fringe populations with high baseline Rh-negative frequencies, for reasons that include the systematic introduction of European genetic material into African American gene pools through the violence of the plantation system, for reasons that include the founder effect operating in dozens of isolated communities across the American South and Appalachian Mountains, concentrating rare genetic traits in communities that were isolated by geography and by the social exclusion that American racial politics imposed on mixed-race peoples, for reasons that include the genetic legacy of racial passing, millions of individuals carrying their full genetic complexity across the color line into communities that thought they were genetically bounded but were not, for reasons that may include pre-Columbian contact histories that mainstream archaeology has not fully investigated and that ancient DNA research is only beginning to shed light on, and for reasons that extend back before any specifically American history, into the Ice Age genetics of Atlantic fringe populations and the deep time bottlenecks that shape the distribution of genetic variants in ancestral human populations. All of these reasons are darker in various ways and to various degrees. They reflect violence and isolation and suppression and mystery. They point to parts of American history that the official narrative has preferred not to look at directly. They connect living people to ancestors whose stories are encoded in the blood, even when they cannot be found in any document. And they are all at the same time part of the larger story of what it means to be human, which is always a story of movement and mixing and survival and the occasional near extinction and the incredible persistence of life forward through every obstacle that would stop it. America is a particularly concentrated and particularly darker version of that story. But it is the same story that every human population tells, written with different specific events and different specific genetics, but shaped by the same fundamental forces. In the end, The blood is not a mystery to be solved. It is a record to be read, partially, imperfectly, with humility about what we do not yet know and patience about what the future might reveal, and with honesty about what the reading already shows, even when what it shows is uncomfortable and dark and far more complicated than the simplified stories we were told. This is the America that the blood describes. Not the one in the textbooks, the real one. The one that existed before anyone wrote it down. And the one that the written record tried to describe and partially succeeded and partially failed. And the one that continues to exist in the genetics of millions of living people who are only beginning to have access to the tools that let them understand what they carry and why. It is a darker country than the simplified story suggests, and a more interesting one, and a more human one, because the complicated, dark, genuinely mysterious story is always more human than the clean version. Because the human story is always about survival through circumstances that should not have been survived. About the mixing of peoples and genetics across the artificial barriers that any particular moment's social order tries to maintain. About the persistence of the body through the attempts of history to destroy or categorize or control it. The blood persists forward into the future. It carries what happened backward through the past. And in the present, in the millions of American bodies where it moves right now, it holds both the darkness of how it came to be here and the wonder of the fact that it is here at all. That is the American RH negative story. Incomplete, as all honest stories are. Dark, as all true stories about how human populations are shaped tend to be. And worth telling, worth following through its long complexity and its genuine uncertainties. Worth sitting within the dark until the first suggestion of something like understanding begins to come through. The journey has been long. You have stayed with it through all the layered history and all the careful uncertainty and all the places where the evidence gives out and the speculation begins. And now here we are at the end of a night's traveling, in the particular stillness that comes after a long effort. In a moment, or in many moments because the body finds its own pace towards sleep, the story will recede and the dark will be just dark and the body will settle into its own rhythms. The heart moving the blood through all its circuits. The cells doing their ancient work. The genetics sitting quietly in the nucleus of every cell, carrying its record of everything that happened on the way here. You carry that record too, whatever your blood type, whatever your ancestry, whatever the story of how your particular lineage arrived at this particular moment in this particular body. You are the current end of an unbroken chain of survival that stretches back through every dark thing that happened to the people who preceded you, and through every miracle of persistence that allowed them to survive those dark things and pass life forward. That is what you carry. That is what the blood is. Not a mystery to be solved, or a mark of distinction, or a curse, or a blessing in any simple sense. Just the accumulated record of an unbroken line of surviving, moving through you right now as you settle into the dark, as the story fades, as the night does what nights do. Rest now. The blood keeps moving even when you are still. The record keeps existing even when you are not thinking about it. The history is there whether you know it or not, whether the country that contains you acknowledges it or not. It is enough that you listened, that you followed the question honestly into the dark places it led, that you sat with the complexity and the uncertainty and the grief and the wonder that an honest engagement with this material produces. The blood carries the history, and you for one night let the history be told. If this story kept you company through the dark hours, if the mystery of the blood gave your mind somewhere to wander while the body found its way toward rest, then it did what it was made to do. You can leave a comment if you want to, just something quiet about where you are in the world right now and what the night looks like from where you are sitting. Someone in the mountains of Appalachia, someone in a city apartment, someone in a place far from America listening to a story about a country whose blood carries more complexity than most people know. All of us in the dark together, connected by the fact of being human and by the questions that being human generates about where we came from and what the blood remembers and what the past has to say to the present. Rest well. The blood keeps its record in the dark as in the light, and the story will still be there in the morning, waiting for anyone who wants to follow it further. There is something that happens in the hours between midnight and dawn that does not happen at any other time of day. The mind loosens its grip on what it is sure about. The categories that feel solid in daylight become softer. The boundaries between what is known and what is merely believed, between what happened and what might have happened, become more permeable. And in that permeability, something like genuine curiosity becomes possible. The kind of curiosity that does not need to arrive immediately at an answer, that is willing to sit with the question as long as the question deserves. This story has been trying to inhabit that space, the space where the answer to why America carries unusual amounts of Rh-negative blood is not one clean thing, but a layered complexity. A convergence of colonial history and forced genetic mixing and isolated communities and the founder effect and racial passing and pre-Columbian mysteries and deep-time population genetics and the specific human tragedies that played out in places and in bodies that official history did not think worth documenting. The blood was there for all of it, moving through bodies that were enslaved and bodies that were free, through bodies isolated in mountain hollows and bodies that crossed racial lines to find safety and new identities, through bodies that carried the genetic legacy of the Atlantic fringe of Europe and bodies that carried the very different genetic legacy of West Africa, and bodies that carried whatever was here before any of the documented history began. And it is here now in bodies that are alive tonight reading or listening, carrying the full weight and complexity of everything that produced them without necessarily having any way to know what that weight is or where it came from. That is the final thing worth sitting with. Not the specific genetics, not the specific history, but the simple extraordinary fact that the people alive right now are the products of everything that happened before them. And that everything that happened before them is written in them. In the genetics, in the epigenetics, in the culture and the memory, and the ways that bodies learn to carry what happened and pass it forward. You are the current end of that forward passing. Whatever you carry, however complicated or simple or dark or luminous your ancestral history, you are arriving in the present, the latest chapter in a story that has been going on for as long as there have been humans to tell it. That is something worth knowing as the night deepens and the body finds its way toward rest. Not a comfort exactly, but something larger than comfort. Something like the particular solace that comes from understanding where you are in a very long story and knowing that the story does not end with you. That it will continue to be carried forward into futures you will not see by people whose bodies will hold everything you carry and more. The blood moves forward. The record persists. And you, right now, in the dark, are part of what it is carrying. Also, like and subscribe to my channel. Ancient Tales Sleep US Secrets. Good night, sweet dreams.